Yep.
So thank you everyone for making your early morning available, or afternoon or evening, depending on which part of the world you are in, to join us. And Vivek, you could welcome the audience as well as the speakers, and then we will start the meeting with your welcome.
All right. Good morning, all. For those in the East Coast and the US, and I see in the chat box, we have a global audience. I see audience from Riyadh, from Nigeria, from Belgium, from India, and from UK. And, I'm from Belgium. And thank you so much for everyone for, participating in this. And, one of the main things is, we wanted to organize this workshop because we wanted to do it live in person, but, in these times, we moved it as a virtual meeting, but it's great to have a virtual meeting because we can have this as a global audience, global talk. We see people from Australia as well joining in, from Brazil. Wow, it's truly an Iran, and it's truly a global audience. As we all know, cancer is a disease of the genes. With the advent of precision medicine over the last decade, there has been an explosive growth in the availability of so many techniques. And genomic testing is emerging to be a part of the routine oncology care. And given its rapid evolution and decreasing costs, it's quickly emerging to being essential. And this symposium brings together pioneers in this area to help understand the cutting-edge science and promote knowledge in the next decade in precision medicine. And thank you all for dialing in and joining in from all over the world. We are thrilled and delighted to kick off this call. Dr. Mani, the floor is all yours.
Okay. So I
just introduce Dr. Mani. Let me go ahead and introduce Dr. Mani so that set the stage as well. So hope you all can see my screen. Right. Welcome to the Precision Oncology, the Next Generation workshop. So Dr. Mani is a professor at MD Anderson Cancer Center and in the Department of Translational Molecular Pathology. He's a co-director of the Metastatic Research Center, co-director for Stem Cell and Developmental Biology in the University of Texas, MD Anderson Cancer Center. So as you may all know, as well recognized, he was a postdoc with Dr. Bob Weinberg in the Whitehead Institute and MIT. He discovered for the first time that cancer cells can actively activate latent EMT, the embryonic epithelial mesenchymal program, and become stem cells. And again, this work has been cited over, I think, if you take everything together, it's over 10,000 times. And very few papers get this kind of attention. And this was, in fact, featured in the New York Times, Boston Globe, and many other magazines and all over national media. And this made him a world famous. And it's a pleasure and honor to welcome him. In addition, he's been elected as a fellow of the American Association of Advancement of Science, a full member of the Sigma C, the Scientific Research Honor Society, American Cancer Society Research Scholar, V Foundation's V Scholar. And again, the thing is that he's a basic scientist, and he always, thinks about, translation, thinks about, translation. So, I never saw Dr. Mani before going to India. So, we were in a meeting in India, and I happened to be a speaker there, and he was a speaker there as well. We introduced each other. And I said, I worked at MD Anderson, and he also said he worked at MD Anderson. So that's how we got to know each other. And, he was always wanting to do translational science and trying to help patients. And recently, he has even co-founded a precision oncology company called Ilon Precision Oncology Company. So over to Dr. Mani to take over the stage.
Thank you, Vivek. As Vivek said, we both work at MD Anderson, but we have to meet in Chennai at a different meeting to get to know each other and started working together in various aspects. And again, once again, I want to welcome all of you and thanks all of you for joining in. Special thanks to Ebony because she makes this seamlessly possible. Thank you, Ebony. And just have a few slides. Again, the goal, I have been hosting these kind of meetings, many of them. The simple reason is only one, that, it's very important that education is the key. So therefore, just the focus is education. And if you notice, the theme of this meeting is also an education. So feel free to post questions in the chat window, not in the chat window, the Q&A window, and we will read it out. We will also make sure to say your name. So therefore, this is more of, again, not only just be a scientific talk, but also an educational conversation. So feel free to ask anything you would like to ask from these experts who are really pioneers in this area. Once again, I thank Vivek for, he has done a remarkable work in this area. One of the questions which somebody asked me is, why am I organizing with Dr. Vivek Sobeja this meeting on precision oncology, which I don't work on. In fact, that's wrong, because as you can see, this is again a slide which you will hear from many people. What is the current medicine, how that works, and it benefits some people, but not for everyone. Through precision medicine, you can make a positive impact for many patients. As I was mentioning, how am I involved, and how every one of you who's dialing in or who's not dialing in involved. If you think about, if you look at a biology, everything, particularly cancer biology, we all work towards one goal, which is find a way to treat patients. So this is kind of, the patient, you can use a patient sample, cell line, human directly in-person model, et cetera, et cetera. Ultimately, that's all we are working towards. This slide kind of shows that, one of the things in drug development is that, you are developing a drug and you're also developing a biomarker. When you're trying to develop a biomarker, you're also trying to understand the biology. So almost every one of us is working in a precision oncology, but we don't call that way. That's all, in my opinion. So, in other words, the goal is kind of integrating all these various information and then coming up with the personalized precision oncology.
I think you should move to a presenter mode, because we see you switch to the display mode.
Oh, sorry about that. Thank you for.
Go to the display settings and you can change that. Display mode.
Display mode. Sorry. There's a way I can swap screen.
If you go up, there's a display settings. The next one.
The next one here.
It says display settings.
Sorry, here. No. Sorry. The Q&A, new share, pause, and it.
That's fine. I think this is good enough. Yeah, go ahead.
Sorry, I'll. There's a way I can swap the slide, but it's not.
No worries. No worries. That's fine. This is good enough.
Sorry about that. Here.
Hide presenter view.
Sorry.
Go up to that thing. Hide presenter view. Yeah.
Oh, thanks a lot. Okay, thank you. Appreciate it. So, what we are trying to do is we are trying to integrate these various information and understand how that kind of, precision oncology, and you're personalizing the precision oncology. So, when MD Anderson started a personalized oncology a while ago, when Massimo Cristofanelli, my good friend, was here, they were also working on precision oncology. Now we understand that precision oncology could be personalized. And that's kind of theme of today's this meeting. Again, I'm honored to host this meeting with Vivek Sobeja. We speak same language in addition to English. And he's, again, I have to meet him in Chennai to get to know him more, even though we work in the same place. And he's an associate professor at the Department of Investigational Cancer Therapeutics. He's also the medical director for Center for Targeted Therapy. He's an executive director for Cancer Medicine Research. and he's also an associate professor at Pediatrics, Division of Pediatrics. He has conducted many clinical trials, over 50, and he's one person are very few who are both adult and pediatric oncologists. He leads several inhuman and practice-changing novel basket trials. You will hear more from Vivek later. And then he's also PI of rare oncology agonistic research, or ROR trial. And he helped to lead the BRAF-MEK Fda approval for the treatment of BRAF V600 mutant anaplastic thyroid cancer. This is the first Fda approval for this cancer. He's, as you all know, he's a major advocate for precision oncology, and he's also global co-PI for this sorry, I cannot say this name, Fda approval in Erdheim-Chester disease. And he's a global co-PI for 2 selective RET inhibitors. Probably you all must have seen the news recently, and there are many other drugs. He helped to bring them to Fda approval through various clinical trials. And he's a PI in the trials leading to approval of Rubinectin. So you can read it. He's an RO-funded investigator, and he's a PI for a non-BRAF V100 arm of NCA match. And he published a lot of papers, and including the recent New England Journal of Medicine, Journal of Clinical Oncology, Cancer Discovery and Cell. He received an award, Presidential Excellence Award from MD Anderson. He's also recently been elected as a fellow of Sabin Family Foundation. So, it's an honor, and I'm sure those of you who are following Twitter, he's one of the top 20 oncology influencers in Twitter, Q2 2021, some website. They've analyzed the data, and here is Vivek. So again, what we are trying to do is, one other thing we are working with Vivek, trying to understand, dissect intrinsic resistance from an acquired resistance, where, how these cells can either intrinsically resistant or acquire resistance for a period of time, in particularly the RET inhibitor scenario. And we are performing a single cell RNA-seq in my lab, and at various times point, hoping to understand at a single cell level the intrinsic resistance and acquired resistance, and then how to target them. And in my lab, we study a program called EMT, as Vivek said, and then its interaction with the tumor microenvironment as well as immune microenvironment. Here is my group, and you can find more information at money.us. Without further ado, I would transfer the podium to Vivek to introduce our first speaker. And once again, I thank you all for making time to join us.
All right, outstanding. So going on, it's a pleasure and honor to welcome Dr. Nicole Resner. She's a medical officer in the US FDA. Dr. Nicole Resner completed her medical degree at the St. George's University School of Medicine. She completed a pediatric residency at the Cohen Children's Medical Center of New York, and then completed her pediatric hematology oncology fellowship at Children's National Hospital. She remained at the Children's National Hospital for an additional year, and as a pediatric neuro-oncology fellow. She joined the thoracic and head and neck oncology team in the Division of Oncology 2 in the FDA as a medical officer in July 2016. And she became the team lead of thoracic and head and neck team in August 2020. As we all know, thoracic, especially lung cancer, has become the poster child for precision oncology. And I'm sure her work as a lead in the Division of Oncology 2 has saved so many people's lives in the real world. And thank you, Dr. Drezner. I also found out just a couple of days ago that she also did medical school with my wife, and they were training together in New York hospitals. So it's a small world. Thank you so much for agreeing to accept our invite, and the floor is all yours, Dr. Drezner. Thank you.
Thank you so much. Can you hear me?
Yes, we can.
Perfect. All right, I'll share my screen. All right, great. Thank you so much for having me. My name is Nicole Drezner. As Dr. Sobeja just said, I'm an oncologist at the FDA, where I lead one of the thoracic and head and neck cancer teams. I have no financial relationships to disclose, and I will not discuss off-label or investigational use of FDA-approved therapies. I'm so happy to be here and to be starting off what promises to be a really interesting day with great speakers ahead to discuss a topic that is shaping cancer detection and treatment in increasingly significant ways. I will give a brief overview of precision medicine, go over regulatory considerations for the approval of targeted therapies, focusing mainly on the accelerated approval pathway with thoracic oncology as an example. I'll talk about regulatory considerations for the development and design of clinical trials for targeted therapies. And finally, I will discuss some of the FDA's Oncology Center for Excellence, or OCE, initiatives in precision medicine that aim to move the field forward in collaboration with divisions within the FDA and with multiple different stakeholders. Thanks to an increasing understanding in the molecular pathophysiology and the mechanism of actions of drugs, as well as a deepening knowledge of the many factors that drive a cell to grow into a cancer, we are beginning to use precision medicine in oncology to achieve the goal of targeting the right treatments to the right patients at the right time. Therapies that act on specific molecular targets that are associated with a particular cancer or group of cancers have revolutionized many of the treatment paradigms we have depended on for years, in some cases allowing patients to entirely forego the global cytotoxicity of conventional chemotherapy. The scientific advances that have paved the way for personalized medicine have required regulatory changes at the FDA as well, through the implementation of programs to expedite drug development and to address the rapid innovation at the bench and in the clinic that has been accelerating over the past 10 years. Thoracic oncology, particularly the field of non-small cell lung cancer, has in some ways emerged as a paradigm for the success of precision medicine, as it has been applied to one of the most common types of cancers in the US that results in the most cancer-related deaths. Beginning in the late 1990s and early 2000s with the sequencing of the human genome, followed by the first cancer genome, tyrosine kinase inhibitors targeting gain-of-function oncogenic mutations elicited dramatic responses in some cancers. Although the first successful kinase inhibitor, imatinib, which was approved for the treatment of CML in 2001, was approved in 2001, development in lung cancer began soon after. Shown here is a timeline of FDA approvals of targeted therapies for the treatment of patients with lung cancer, beginning with gefitinib, approved in 2003 for the treatment of EGFR mutated non-small cell lung cancer. Since then, there have been almost 30 drugs approved that are active against a wide range of targets in lung cancer, from PD-L1 to RET. A necessary companion to the development and approval of targeted therapies is the need for timely and accurate molecular profiling. Technologic advances in PCR and next-generation sequencing have paved the way for a large number of genomic alterations to be identified through parallel sequencing. Many of the therapies shown here were developed and approved with a companion diagnostic for the safe and effective use of the drug product. A 2020 article in a New England Journal of Medicine by Howlatter et al. Described the trends in lung cancer mortality in the US over the past 20 years. As shown in the graphs here, while the incidence of non-small cell lung cancer has declined over that time frame, mortality from non-small cell lung cancer has declined even faster, with a rapid decrease beginning in 2013, coinciding with the year of the first approval of an EGFR TKI, erlotinib, in the first-line setting. The left panel shows the incidence of non-small cell lung cancer in men declining by approximately 3.1% between the years 2008 and 2016, represented by the blue line. However, from 2013 to 2016, the incidence-based mortality fell by 6.3% in red. A similar trend was observed in women on the left, with the incidence of non-small cell lung cancer declining by 1.5% between the years 2006 and 2016, and incidence-based mortality falling by 5.9% between the years 2014 and 2016. Lung cancer-specific survival has improved from 26% of men diagnosed with non-small cell lung cancer in 2001 to 35% of men diagnosed with non-small cell lung cancer in 2014. This effect was observed across all races and ethnic groups, as well as in women. The authors postulated that the reduction in incidence of non-small cell lung cancer, coupled with the treatment advances, which have been driven by the widespread approvals of targeted therapies, are responsible for the decline in mortality from non-small cell lung cancer observed over the past 20 years. Accelerated approval regulations were instituted by the FDA in 1992 to allow for faster approval of drugs for serious conditions that fulfilled an unmet need. These regulations were initially applied to HIV therapies that drastically altered the course of the AIDS epidemic in the US and are now largely used for the approval of oncology drugs. Given that patients with oncogenic mutations may have extended survival with the use of targeted therapy, the accelerated approval pathway is particularly useful in this setting to approve promising drugs faster. Many targeted therapies were first granted accelerated approval by demonstrating a meaningful advantage over available therapy through an effect on an intermediate endpoint reasonably likely to predict clinical benefit. Products approved under the accelerated approval pathway must meet the same statutory standard for safety and effectiveness as those granted traditional approval, and post-marketing trials are required to verify and describe the clinical benefit. Between the years of 2010 and 2019, 82 of the 98 accelerated approvals by the FDA were for oncology indications. The accelerated approval pathway has been particularly successful in facilitating the development of therapies for non-small cell lung cancer. 11 products have been granted accelerated approval for non-small cell lung cancer based on an improvement in overall response rate since 2010. Most of these approvals were for practice-changing targeted therapies such as osimertinib, alectinib, and selpercatinib, although immune checkpoint inhibitors such as pembrolizumab were also approved through the accelerated approval pathway. Confirmatory trials leading to regular approvals of these drugs followed 1 to 3 years after the initial approval. Without this regulatory initiative, patients would have had to wait years after early signs of clinical benefit to have access to novel therapies. One of the reasons why so many targeted therapies for non-small cell lung cancer have been granted accelerated approval is that objective response rate has been established as an intermediate endpoint reasonably likely to predict clinical benefit in this disease setting. In FDA analyses published in 2015, a strong trial-level association was found between ORR and PFS in studies of patients with advanced non-small cell lung cancer dating back to 2003. Although no trial-level association was found between ORR and overall survival, or between PFS and OS, in a patient-level responder analysis, patients who achieved a response had better PFS and OS compared to non-responders. The lack of a trial-level association between ORR and OS was likely due to crossover and longer survival after progression in the targeted therapy and first-line trials included in the analysis. This slide divides the FDA-approved therapies for advanced non-small cell lung cancer between 2010 and 2021 by the endpoint leading to their initial registration. As you can see, a large number of targeted therapies were approved based on an improvement in ORR. Most of the earlier approved therapies have since had a subsequent confirmatory trial demonstrating a PFS benefit. Of note, crizotinib and entrectinib and the combination of dabrafenib and trametinib for the treatment of ROS1 and BRAF V600E mutant non-small cell lung cancer, respectively, were granted regular approval based on response rate for several reasons. The large magnitude of improvement observed in ORR over available therapies, the durability of the responses, and the rarity of the mutations, making randomized trials difficult to conduct, especially given the ORR and DOR results with the targeted therapy. This brings us to some of the regulatory challenges in reviewing applications for targeted therapies for accelerated or regular approval that I alluded to in the prior slide. Smaller subgroups of patients defined by molecular alterations make it difficult to design trials that are powered to demonstrate an effect on OS due to the size of the trial that would be required. This issue was raised during the review and approval of selpercatinib for RET-positive non-small cell lung cancer. Second, while the traditional gold standard endpoint for oncology is overall survival, most regular approvals of targeted therapies were granted on the basis of an improvement in PFS. The regular approval of alectinib for the treatment of patients with ALK-positive non-small cell lung cancer illustrated the adequacy of a dramatic improvement in PFS over another targeted therapy to support registration. Finally, the different safety profiles and route of administration of targeted therapies versus available therapies are characteristics that must be considered in a risk-benefit assessment of the application, as seen in the accelerated approval of tepotinib for patients with non-small cell lung cancer with MET exon 14 skipping mutations. The accelerated approval of selpercatinib for patients with RET-positive non-small cell lung cancer in May 2020 was a line agnostic approval based on robust and durable ORRs of 64% and 85% in patients who were previously treated and who were treatment naive, respectively. The FDA review team concluded that the ORR and DOR were sufficient to establish the clinical benefit of selpercatinib in this rare subgroup of patients, comprising approximately 1 to 2% of patients with metastatic non-small cell lung cancer. And so rather than a confirmatory randomized trial, a post-marketing requirement was issued for data from additional patients, both treatment naive and previously treated, to confirm the clinical benefit. Furthermore, given the robust ORRs observed in patients receiving selpercatinib in the trial supporting accelerated approval, equipoise in trial design may be compromised as patients would need to be randomized to a likely inferior comparator. Similar to selpercatinib, high response rates were observed in the early single-arm studies of alectinib in previously treated patients with ALK-positive non-small cell lung cancer, which led to its accelerated approval in 2015. In the pivotal trial supporting regular approval, in which patients were randomized to receive alectinib or crizotinib, the standard of care prior to the development of alectinib, crossover was permitted for patients on the crizotinib arm with disease progression. Allowance of crossover has become common practice in trials of targeted therapies, and high rates of crossover may confound the assessment of the OS analyses, which are typically event-driven and may take place years after the primary PFS analysis has read out. In the case of alectinib, which received regular approval in November 2017, OS data was still immature as of an update presented at ASCO in 2020, though it maintained its survival benefit over crizotinib. In the case of tepotinib, which received a line-agnostic accelerated approval for the treatment of patients with non-small cell lung cancer with MET exon 14 skipping alterations in February 2021, the ORR of 43% observed in the treatment-naive patients in the primary efficacy population is in the range of that observed with FDA-approved available therapies. Standard treatment options for these patients are the same as those used for patients with non-small cell lung cancer without a specific driver mutation, that is, platinum-based chemotherapy and/or an anti-PD-L1 antibody. The highest ORRs observed for these therapies ranges from 48 to 58% in patients receiving pembrolizumab or atezolizumab in combination with chemotherapy. However, given tepotinib's differing safety profile and the option for a treatment with a single agent administered orally, the FDA review team concluded that the risk-benefit profile still favored tepotinib as a valuable treatment option for the treatment-naive population. I will now turn to a discussion on regulatory considerations for the design of clinical trials intended to support marketing applications for targeted therapies, focusing on master protocols, early biomarkers and their inclusion in study design, and decentralized trials. The era of genomic medicine has required changes in the way that clinical trials are conceived and implemented, building in more efficiency than the classic phase 1 to 2 to 3 pathway, and using the emerging science of prognostic molecular biomarkers such as ctDNA to create new endpoints and improved risk stratification. The master protocol framework has supported the approval of several targeted therapies for tissue-agnostic indications, predicated on the idea that cancers arise from common somatic genetic building blocks that may be targeted by one therapy, regardless of tumor type. Decentralized trials provide another mechanism for reaching patients, especially those who may not live within an academic center's catchment area, for trial recruitment, and are particularly useful in testing therapies for populations of patients affected by rare mutations. Master protocols are intended to expedite late-stage drug development through trial designs that test multiple drugs and/or multiple cancer subpopulations in parallel under a single trial framework. Through a single infrastructure, trial design, and protocol, master protocols can simultaneously evaluate multiple drugs and/or disease populations, allowing for more efficient drug development. In general, there are two major types of master protocol design: basket trials and umbrella trials, with sample schemas shown here. A master protocol designed to test a single investigational drug or drug combination in different populations is commonly referred to as a basket trial, shown on the left. Sub-studies within basket trials are usually designed as single-arm activity estimating trials with ORR as the primary endpoint. A strong response signal within a substudy may allow for expansion of the substudy under the master protocol to generate data that could potentially support a marketing application. Umbrella trials, shown on the right, are designed to evaluate multiple investigational drugs administered as single agents or in combinations in a single disease population. Substudies within umbrella trials can include dose-finding components to identify safe doses of an investigational drug combination before proceeding with the activity estimating component. Umbrella trials can also employ randomized controlled designs to compare the activity of the investigational drugs with a common control arm. Shown here are descriptions of the 5 drugs that have received accelerated approval for tissue-agnostic indications based on the presence of a molecular alteration: microsatellite instability, deficient mismatch repair, NTRK gene fusions, and high tumor mutational burden. Each approval was based on results from a relatively small efficacy population derived from multiple single-arm cohorts or as part of a basket trial design. The determination of whether a drug is suitable for tissue-agnostic development depends on several preclinical and clinical factors. Generally, tissue-agnostic development can be considered when a drug-target combination demonstrates high activity across multiple tumor types. Other developmental considerations include differences in natural history across diverse cancers and how data can be obtained from enough patients across different tumor types. The difficulty in identifying rare genomic mutations through testing is also a major issue in tissue-agnostic development. Next-generation sequencing platforms for routine clinical use can simultaneously sequence many cancer genes at once, but these assays may only be used routinely in a small number of major academic centers. Although most of the approvals listed here included prospective or retrospective central testing, this may be cost prohibitive in some cases. A final risk in tissue-agnostic development involves the possibility that the targeted therapy could be ineffective for a specific tumor type despite the presence of the genomic alteration due to either the development of resistance mutations or other co-occurring oncogenic driver mutations. A strong biologic rationale, supportive preclinical data, and demonstration of consistency of responses across tumor types are generally necessary to mitigate this risk. Each of the therapies listed here have associated post-marketing requirements for additional data demonstrating the effect of the drug in a variety of tumor types. Other biomarkers, such as ctDNA, have prognostic value and are being investigated in a variety of different clinical scenarios. Use of ctDNA is being explored at almost every stage of cancer development, from cancer detection through screening to molecular profiling as a prognostic indicator, detecting residual disease after an initial therapy, and to monitoring response of therapy as well as clonal evolution and recurrence. Here I will focus on using ctDNA to detect minimal residual disease, or MRD, after definitive therapy such as surgery. Shown here are some of the data for ctDNA as a prognostic biomarker in early stage lung cancer, breast cancer, bladder cancer, and colon cancer. The presence of ctDNA after surgical resection was associated with worse relapse-free survival across these tumor types. 2 limitations of these studies are the inclusion of only small numbers of patients, ranging from 24 to 96 patients per study, and that different technologies were used to assess for ctDNA. However, this data suggests that ctDNA post-surgery has prognostic implications and supports the incorporation of ctDNA as a prognostic biomarker in prospective clinical trials. Clinical trial design using ctDNA as a prognostic biomarker is shown here. A baseline ctDNA sample, or liquid biopsy, is obtained prior to neoadjuvant therapy or surgery. Another liquid biopsy is obtained after surgery, and patients are stratified by biomarker status, such as the presence or absence of ctDNA. Patients would then be randomized to receive standard of care with or without investigational therapy. Following adjuvant treatment, another liquid biopsy could be used to assess the response to therapy, and patients would continue to be followed for disease-free survival and overall survival. This design allows for the efficacy of the investigational drug to be analyzed for the total population or for the biomarker positive group of patients, and can help develop ctDNA or other prognostic biomarkers as early endpoints that could potentially support marketing applications once they are established as being predictive of an improvement in DFS or OS. An enrichment strategy to increase the efficiency of drug development is to only study the investigational adjuvant therapy in the group of patients who are biomarker positive, as only a higher risk population is studied within this design, The DFS and OS event rates would likely be higher and allow for a smaller sample size and shorter trial. A third potential trial design is to obtain liquid biopsies after surgery and at the completion of standard adjuvant therapy. Patients with biomarker-positive disease after adjuvant therapy would be randomized to an investigational therapy or observation and then followed for survival. Mutation testing to assess for mechanism of drug resistance may also be incorporated into this type of design, which may inform the selection of the investigational therapy. There are several points to highlight when drawing parallels between MRD and hematologic malignancy and ctDNA in solid tumors. In hematologic malignancies, MRD is recognized as a prognostic biomarker, although evidence varies across tumor types and patient populations. Cellular and molecular technologies, such as multiparametric flow cytometry and next-generation sequencing, have been shown to reliably quantitate tumor burden. And finally, since 2012, there have been numerous FDA co-sponsored public workshops evaluating MRD as an early endpoint across various hematologic malignancies. As a result of these workshops, FDA issued a guidance for industry for the use of MRD in drug development. For solid tumors, small data sets suggest that ctDNA may also be prognostic for patients with both early stage and advanced stage disease. However, the timing of sampling and quantification methods have not yet been standardized and best practices have not been established. There are ongoing efforts to harmonize ctDNA data to characterize its associations with clinical outcomes and to assess whether trends in smaller data sets can be replicated in larger data sets and prospective trials. Decentralized trials are another innovative way in which precision medicine can be facilitated through trial design. Use of full or hybrid decentralized trial designs by commercial sponsors has been rare in oncology, in part due to uncertainty surrounding the effect of remote assessments on data quality and outcomes. However, the COVID-19 pandemic has resulted in modifications to the structure of ongoing and planned trials due to travel and other restrictions. This development has important implications for precision medicine, as there is an increasing role for biomarker-enriched trials in rare molecular subsets. The decentralization of portions of a clinical trial structure may increase trial access to patients who live far from specialized centers and would otherwise not be included in a clinical trial. Increasing patient recruitment may result in an improvement in the generalizability of study results, especially when the subgroup with the biomarker is small. Some risks to this approach include the need for a standardized schedule of activities and imaging time points across widely disparate study sites in order to maintain study integrity and accurately assess endpoints. Management of serious adverse events may produce a challenge depending on the level of involvement of the local oncologist and access to hospital resources. Oral therapies that can be shipped to the patient are better suited for decentralized trials than IV therapies, which may still require travel. Finally, first-in-human studies are likely not well suited to the decentralized trial structure given the number of safety and pharmacokinetic assessments that are required to characterize the safety of the study drug. Nevertheless, the decentralized trial design has the potential to facilitate much wider access of targeted therapies to patients. The OCE is now requesting that applicants submitting data in support of NDAs or BLAs voluntarily add flags to data sets to discriminate between remote assessments and trial site assessments. Learning from trials conducted during the COVID-19 pandemic that permitted some aspects of the study to be performed remotely to reduce potential COVID-19 exposure may translate into guidance and collaboration for future decentralized trial development in oncology. Finally, I will discuss the OCE's role in developing precision medicine in collaboration with the oncology community and one of our initiatives to make therapies available globally. The OCE was established in 2017 to streamline the development of cancer therapies by uniting experts from FDA product centers to conduct expedited reviews of drugs, biologics, and devices. There was a recognition that novel endpoints are needed to describe the effect of targeted therapies and that complementary and companion diagnostics will be increasingly used to inform treatment decisions. These both require multidisciplinary input across all the hematology and oncology divisions in the Center for Drug Evaluation and Research, as well as experts in the Center for Devices and Radiological Health and the Center for Biologics Evaluation and Research. The OCE enables an integration of resources at the FDA to facilitate the transition of targeted therapy from tumor site-based to gene or pathway-based approaches. However, oncology drug development also requires global collaboration, with pivotal trials increasingly being conducted internationally. Despite the international study population that support the marketing applications for many drugs and biologics that are submitted to the FDA for review, there are often significant delays in submissions to other regulatory health authorities. These delayed submissions may lead to delayed approvals and drug access for patients outside the US. A major consequence of delayed approvals of new therapies is the development of dissimilar standards of care between countries, which may limit eligibility of patients and thereby impede trial enrollment from some geographic regions. Project Orbis was developed through existing global connections between international health regulatory agents in 2019, paving the way for direct collaboration between the agencies during the review of concurrently submitted marketing applications. Project Orbis reviews typically focus on high impact and clinically significant applications. In thoracic oncology, 7 targeted therapies have been approved by the FDA under Project Orbis. These include a novel combination therapy, the first adjuvant targeted therapy for early stage lung cancer, the first FDA approval for malignant pleural mesothelioma in over 15 years, and 4 new molecular entities. Additionally, early data suggests that the implementation of Project Orbis has reduced the approval gap between the FDA and other regulatory agencies across the world, bringing these targeted therapies to patients sooner. In conclusion, through innovative trial designs and the effective use of accelerated approvals, targeted therapies have become available sooner, both in the US and globally. The widespread use of practice-changing therapeutics, largely targeted therapies and immunotherapies, may contribute to declining mortality rates, with data from non-small cell lung cancer providing supportive evidence of this. There are multiple OCE initiatives to support the development of precision oncology, including Project Orbis, which has accelerated the timeline of worldwide approvals of high-impact, practice-changing therapies and broadened the scope of precision medicine across the world. Thank you so much for listening, and I would like to acknowledge and thank my colleagues at the FDA who have all contributed to this presentation and the ideas I've shared. Thank you.
Thank you so much, Dr. Drezner. I think we have one question from the audience. That was a fantastic educational talk. And I think a minute ago, New York Times says the FDA grants full approval to Pfizer COVID vaccine. I think this is a historic moment. That's great. Thank you.
Great to hear.
For all what you do in the FDA. I think this is fantastic. I think the COVID vaccine is precision medicine. I think it's personalized precision medicine targeted towards a virus. Thank you all so much in the FDA on what you do. This is a truly historic moment. So one of the questions the audience has is, what are some of the major criteria which the FDA follows before approving a test as a companion diagnostics?
Yeah, so review of a companion diagnostic with a new drug or biologic is done sort of concurrently with the FDA and CDRH, the Center for Devices and Radiological Health, and we're very closely involved in both of our review processes. So the reviews take place separately, but we come together for our meetings and discussions on labeling. Typically, CDRH runs the requirements or sets the requirements for approval of a companion diagnostic. But usually what we want to see is that the companion diagnostic has been used in the pivotal trial supporting approval. And if not, that there's been a bridging study between whatever diagnostic was used in the trial supporting approval and the to-be marketed device. A lot of times we run into issues of local lab testing, which is commonly used in clinical trials. And so usually CDRH wants sponsors to have collected all of the information about the specific local lab tests to a very granular level in order to be able to bridge that data to the to-be marketed companion diagnostic assay. And they have a lot of other requirements that go into testing that I am sort of not an expert in, but we do collaborate with them really closely. And we in the OCE or in Division of Oncology too start giving comments to sponsors to think about device development very early on in development, even in phase 1 trials, we're encouraging people to talk to CDRH so that they can develop their diagnostic.
Oh, fantastic. And yeah, go ahead, Mani.
Thank you again for the wonderful talk. I'm just wondering, this MRD is applicable as a criteria for a clinical approval, primarily for a lung cancer or for any cancer?
I think potentially any cancer. I mean, the issue, is that this is somewhat less well characterized in solid tumors than in hematologic malignancies. And so we're, at least in lung cancer, we've seen clinical trials that have been designed, with some early biomarkers, not necessarily ctDNA, but other things like pathologic complete response, which are starting to emerge as potential early biomarkers that could support earlier approvals. But we really don't have the data yet to say that they are an intermediate endpoint, that potentially support an accelerated approval. And so once we start compiling data in clinical trials that use these earlier biomarkers like ctDNA or PathCR or MRD, and then also are following patients for event-free survival or disease-free survival or overall survival, we can sort of start to make connections between the trial level association, do trial level analyses of the association of these early endpoints with later, more gold standard type endpoints, which I think it will be necessary for us to develop earlier, these earlier endpoints as endpoints that could really support approvals.
Thank you.
Yeah, perfect. Thank you so much. Just one more question, which I had was, pediatrics, right? What would be FDA's consideration for pediatric drug approvals? Because, we saw that the RET inhibitor and several of the drugs are approved 12 and above. So, how long would it take for kids less than 12 years of age?
This is
a question close to all our hearts because, we have small kids and we are looking forward for this question, answer from FDA. How long would it take in oncology, more so in oncology?
I think that the OC is very interested in pediatric development, and the Cures Act has sort of ushered in a period that in which we're really encouraging pediatric development more. Previously, sponsors were able to waive the requirement to conduct pediatric studies just based on the tumor type that they were studying their drug in. And now, as of August 2020, companies who are submitting new molecular entities for as an NDA or a BLA have to do studies, or at least do early clinical studies in pediatric patients if the mechanism of action of their drug is relevant to pediatric cancers. And I think that's really changed the sort of paradigm of how we look at pediatric cancers at the FDA and with sponsors. I mean, having done my training in pediatric oncology, I'm very familiar with the way that clinical trials are run in pediatrics, which is quite different than in adults. And there's really, at least when I was a fellow, far fewer sponsored trials by pharmaceutical companies in pediatrics than there are in adults. And most of the trials, were led through cooperative groups like the Children's Oncology Group. And I think that with this approach to sort of push companies to develop their drugs based on a mechanism of action will really help pediatric drug development. As in terms of an exact timeline, I don't know if I can give you that, but I do know that, we were all really excited, both at the FDA and then in the clinic when I go back to Children's National for the approvals, the tissue agnostic approvals and the things that were approved down to 12 years of age.
Right. So I think that sets the stage for our next speaker. Mani, do you want to go ahead and introduce Dr. Hayes Jarvin?
Sure. And also, there are, Dr. Drezner, there are a number of questions in the Q&A. I'm sorry for, we're trying to keep the presentation on time. And if you could kindly answer those.
I'll go ahead and answer them. Thanks.
I would greatly appreciate it. Again, thanks again to all of you.
Katie Janeway is next. Again, she has a conflict. So let's go ahead and introduce Dr. Katie Janeway. All right. Thank you, Dr. Hayes Jarvin, for your patience and for everything. And let me go ahead and introduce Dr. Janeway. So Dr. Janeway is a pediatric oncologist at Dana-Farber Boston Children's Hospital and Blood Disorders Cancer Center. She's an expert in caring for children, adolescents, and young adults with sarcomas. Dr. Janeway's research is focused on genomics, precision oncology, and bone sarcomas. She leads clinical trials in osteosarcoma and Ewing sarcoma, both as an independent investigator and as the chair of the Children's Oncology Group Bone Tumor Committee. Dr. Janeway's prior work has uncovered genomic events underlying difficult-to-treat childhood malignancies. She has leveraged this understanding to expand treatment opportunities and improve clinical trials for children with advanced solid malignancies. The research group is currently focused on better characterizing the clinical impact of genomic sequencing. The group is also involved in efforts to share and aggregate both sequencing and clinical data to enable a deeper understanding of childhood cancers. We have several data generation and sharing projects aiming to better characterize the genomics of bone tumors like osteosarcoma and Ewing sarcoma, area of highly unmet need in pediatric malignancies. With these projects, Dr. Janeway's group hopes to identify clinical genomic factors explaining the prognosis, treatment, response, and resistance. Dr. Janeway, the floor is all yours.
Thank you so much for that nice introduction and flexibility in the schedule. I really appreciate it. So I'm going to share my screen and enter presenter mode. Please let me know if that doesn't work. Oh dear. Sorry. Okay, can you guys see my screen?
Yeah, we can see your screen.
Okay, thank you. Okay, great. So I'm thrilled to be invited to join you today to discuss precision oncology and childhood cancer. And I'll really address how we're learning from children and from their data. These are my disclosures. I'm a pediatrician, so I will be discussing off-label use. So just to give you an overview of what I'll discuss today, first I'll briefly give you some background on pediatric cancer incidence and outcomes, because many of you probably don't think about pediatric cancer every day. And then I'll cover 3 lessons learned from children and their data. The first is the need to study the precision oncology paradigm in rare pediatric malignancies. The second is real-world data resulting from the study of precision oncology can highlight unmet needs. And then finally, we can use this and other data to improve clinical trial design for efficient study of new drugs and rare cancers. So pediatric cancer obviously is not one cancer type. It's actually a collection of sort of very rare or ultra rare pediatric cancer types, with leukemia, acute leukemia being the most common, representing about 1/4 of diagnoses. But even with leukemia being the most common, there are only 3,000 cases approximately per year in the United States, which actually meets the definition by itself of a rare cancer. But you can see that the rest of the malignancies and the ones that I will focus on here in this talk primarily, which are the pediatric extracranial solid tumors represented by this rainbow of colors over here, are really ultra rare cancer diagnoses. I think, as is familiar to most of you, the outcomes in pediatric cancer, I'm showing survival over over many years, starting in 1975 and ending in about 2008. Outcomes or survival have improved steadily over time. But as we all know, sort of general trends often hide sort of unmet needs underlying those. And so just to show an example of one of our rarer extracranial solid tumor types, osteosarcoma, you can see in this study using SEER data conducted by Lisa Mirabello that if you look at survival curves by decade, there's been little improvement since the mid-1980s. And that's related to the fact that in this rare cancer type, there have been no sort of new approaches to therapy during that time. Already discussed in this conference with the last speaker are next generation sequencing diagnostic tests, which are standard of care for adult solid malignancies. And in fact, even have FDA approval for several of those targeted NGS tests, and then incorporation into NCCN guidelines, for example, for non-small cell lung cancer. But the studies that support the FDA approvals and therefore the incorporation into guidelines are really restricted to those more common adult solid malignancies, which has led us to my group to be quite interested in better understanding the clinical impact of molecular tumor profiling in children. And specifically, do children with pediatric solid malignancies benefit from the precision oncology approach or paradigm? And so I'll tell you about one of the studies my laboratory has been conducting and actually continues to conduct, but for which we are working on an interim report called the GAIN ICAT2 study. And the GAIN study is conducted by a consortium. GAIN stands for Genomic Assessment Informs Novel Therapy, of 12 pediatric cancer groups. I really need to give credit for Alana Church, who is a molecular pathologist who sort of works on this project with me, on this study with me, and she was awarded the pediatric prize at this past ASCO for this interim report. So this GAIN Consortium ICAT2 study is a prospective cohort study for which patients with high-risk extracranial solid tumors who diagnosed at age less than 30 are eligible. By high risk, we mean either already relapsed and refractory or with a 2-year event-free survival, 50% or less. Patients are consented to participate in the study. We use tumor tissue already removed from the patient for clinical purposes, biopsy or resection. We perform either perform next generation sequencing tests with our own laboratory developed test conducted at the Brigham called OncoPanel, and either whole transcriptome or targeted RNA sequencing. Or we accept testing performed outside of our, at an outside laboratory, and we have a limited list of laboratories. We then provide additional clinical interpretation of that NGS testing in what we call an individualized cancer therapy or ICAT recommendation report, and both the test results and that interpretation are returned to the patient and treating oncologist. We collect baseline demographics. We have data on the sequencing results. We obviously follow or collect this sort of clinical interpretation and then look at treatment received and outcome. So the primary aim of the study is to describe overall survival according to whether patients received matched targeted therapy identified by or recommended by tumor profiling. Secondary and exploratory objectives include describing the frequency and range of molecular alterations and in-depth assessment of extraordinary response to matched targeted therapy. For this interim analysis, we planned to describe molecular alterations characterized with regard to impact on diagnosis or response to matched targeted therapy, and the intermediate clinical endpoints to identify extraordinary responders to matched targeted therapy. We included the first 389 patients enrolled, which resulted in 345 patients in an analytic cohort based on all of them having at least one OncoPanel report. We did exclude the outside reports for this interim analysis just for simplicity of analyzing the genomic events. In terms of outcomes for this study, we characterize or classify genomic alterations for potential clinical impact as follows. For diagnostic and prognostic implications, we use AMP, CAP, and ASCO guidelines, which have 2 different tiers. And then for the therapeutic associations, we use an evidence-based tiering system we developed for a predecessor exploratory study because this study was initiated before sort of standard guidelines were available. With tiers 1 and 2 corresponding to clinical evidence, tiers 3 and 4 preclinical evidence, and tier 5 consensus opinion. And then we actually defined extraordinary response to matched targeted therapy as a patient receiving with resist measurable disease at baseline, who had duration of therapy more than 4 months or had a response to monotherapy matched targeted therapy. Baseline characteristics for the patient population in the analytic cohort are age at diagnosis of 11 years and at enrollment of 13 years. And fully 2/3 of the patients had a sarcoma, a rare sarcoma. And there was expected distribution of race and ethnicity according to the sites where patients were enrolled. So looking at this analytic patient population who were eligible for analysis, about 68% of the patients were relapsed or refractory at the time of the data cutoff. 62% of those patients had a molecular alteration for which there was evidence to suggest potential response to match targeted therapy, or in our words, had received an ICAT recommendation. And when we follow the group of patients who received an ICAT recommendation to determine their treatment and response, we had a small subset of patients, 10%, who were not eligible for assessment of receiving match-targeted therapy due to having either incomplete follow-up data or having unfortunately died prior to receiving the molecular report. When we focus on those patients, those 205 patients who were eligible for assessment of receiving match-targeted therapy, the majority of those patients have relapsed and refractory disease, but a subset had newly diagnosed disease. And if we look at the patients who might have been able to receive match-targeted therapy, either because they were getting treatment and actually had a match-targeted therapy that was available to them based on having a formulation that could be taken, and a dose that was known to be able to be given to that patient, 30% of those patients received match-targeted therapy. And when we looked at the patients who received match-targeted therapy, 19% met this very strict definition we developed for having an extraordinary response. Just to show you those extraordinary responders in more detail, the patients sort of spanned the time frame of the enrollment of the study for the analytic population for this interim report. All patients had fusions that activated signaling. There were a range of diagnoses and a range of tiers of ICAT recommendations. And the majority of those patients had at least a partial response. Moving on to look at the or discuss the diagnostic and prognostic impact of molecular tumor profiling for these patients, I'm showing you sort of the bins of diagnoses for the patients in the study here in the center of this circle plot. On the outer circle are the genomic results and whether or not there was an alteration that had either a tier 1 or tier 2 AMP ASCO guideline, met tier 1 or tier 2 AMP ASCO guidelines for a diagnostic alteration. And you can see that fully 60% of the patients had a tier 1 or tier 2 AMP ASCO diagnostic finding, and the majority of those diagnostic findings were gene fusions. Then I just wanted to make one more point, which is to say that you can see that this patient population really consists of we have a long tail of diagnoses. In other words, there are a large number of patients who have sort of ultra rare diagnoses. The final point I want to make about the diagnostic findings in this study is we had 17 patients, mostly with sarcomas, who had alterations identified in our molecular tumor profiling that really changed the diagnostic or clarified the diagnostic classification. This was sort of real time. We sort of had the pathology report prior to our testing and the other pre-enrollment molecular testing that was performed, which often included sort of multiple FISH tests. And occasionally we even saw cases where there was discordance between the sort of gold standard and what we found on NGS. So to summarize the clinical impact and also the outcome after receiving the results, 87% of patients have one or more alteration with potential clinical impact. 30% of those who would have been expected to consider match-targeted therapy received it. 19% of patients receiving match-targeted therapy had this met this very strict definition of extraordinary response, and 5% had a result impacting how their diagnosis was understood compared to the sort of pre-test diagnosis. All responders had a treatment matched to a fusion, and most diagnostically significant alterations were fusions. So the conclusion from this interim analysis is that molecular tumor profiling has a significant impact on diagnosis and treatment recommendations for young patients with solid tumors. And these results emphasize the importance of fusion detection for patients with sarcomas and rare tumors. So I'm excited that the topic of pediatric drug development came up in the questions to the prior talk, and also the topic of COVID vaccine, because I have this slide which sort of addresses both. So this is my COVID vaccine card, and you can see, because I'm a healthcare provider who sees patients, I had my COVID vaccine series completed in January of 2021. These are my children. They are 14 years old, and this is their picture on the day when they received their first COVID vaccine 5 months later. I want to make 2 points about clinical drug development for children with cancer. The first is that based on analysis of PK data and sort of RP2D, recommended phase 2 dose determinations in pediatric phase 1 clinical trials, it has been recognized that for cancer clinical trials, the PK and the maximum tolerated dose for adolescents is almost always the same as what's seen in adults. And therefore, there's actually a recommendation, an FDA guidance, and several sort of position statements recommending that patients 12 and above be included in cancer clinical trials for adults when the histology or molecular alteration is sort of relevant to those patients. And so I wanted to make that first point. But the second point is sort of what came up also in response to the talk, that there are incentives and requirements for trials in children. And the most important one in the context of this conference is what we like to call the RACE Act, which was an update to the Pediatric Research Equity Act, or PREA. And previous to this RACE Act, PREA was indication-based, and adult cancers indications not, so the requirement to study a drug in children was based on whether that indication was seen in children. And because most of the adult indications or cancer types are not, do not occur commonly in children, basically most cancer drugs were exempt from pediatric investigation. But now with this update, if the molecular target being studied is relevant in pediatric cancer, then drugs seeking FDA approval need some type of pediatric study plan. And of course, what that study exactly looks like needs to be tailored to the situation. But what's a bit challenging about this is that the landscape of molecular alterations in pediatric cancers, where we need new treatment approaches, which are the advanced treatment-resistant diseases and rare cancers, is actually not that well understood. So we have 2 pan pediatric cancer sequencing analyses that were published in 2018, and they included about 2,500 patients. But these were primarily sequencing from samples obtained at the time of diagnosis. So looking back at our GAIN-ICAT2 interim patient population, now what we have is a picture of what genomic alterations and what inhibitor drug classes are relevant in the relapsed and refractory patient population. And so I want to make 2 points here. The first is that one thing that's already known and recognized in pediatric oncology is that we have some molecular targets that are unique to pediatric cancers. The perfect example here is EWSR1/FLI1, which previously was not thought to be targetable, but there is a drug currently undergoing phase 1/2 investigation called TK216, which was developed as a direct EWSR1/FLI1 targeting agent. And then the second observation is that we do see pathways altered that are shared with adult cancers. And what comes out of our study is that the RAS-RAF-MAP kinase pathway is one of those pathways. We see mutations across the pathway and indicating potential activity of inhibitors of this pathway. So maybe that's just a fluke of our interim analysis patient population. So to sort of evaluate whether that's the case, we look to the NCI-COG pediatric match trial, of which I am also a part of the steering committee. This is a basket trial designed very similarly to the adult match trial. There is a screening protocol for which patients with relapsed refractory solid tumors are eligible. Biopsies at the time of relapse are sequenced. There's a cassette of multiple different phase 2 clinical trial protocols. There's sort of predefined matching criteria. If you have a predefined matching criteria on the screening protocol, you enter into one of the treatment protocols. And at this last year's ASCO meeting, the first sub-protocol to results was presented, which was of selumetinib, the MEK inhibitor for RAS, RAF, MAP kinase pathway alterations. It accrued 10 patients per year. Here are the different genes for which, in which alterations were found for patients who matched to this sub-protocol. And so we can observe from conducting this prospective cohort study and the basket trial, one area where we need prioritization of molecularly targeted therapies for clinical trials in pediatric cancers. And given that there's a lot of exciting development in agents targeting RAF, RAS, MAP kinase pathway, I just want to put out there, they definitely should have be thinking about a pediatric study plan, which means thinking about a formulation of the drug that can be given to young patients. Finally, we can look at this data emerging from precision studies in children to identify diagnoses that are highly represented in these studies of advanced cancer. So the most common diagnosis, representing 20% of patients in our GAIN-ICAT2 interim population, was osteosarcoma. And I thought, well, that's just because I'm an osteosarcoma expert. But it turns out that's also the most common diagnosis in the pediatric match cohort of the first 1,000 patients. I think we can identify cancer types with poor outcome and limited clinical trial options at recurrence. And for me, that's osteosarcoma. And so just to highlight some ongoing work, we're working on aggregating clinical sequencing data from osteosarcoma patients, both from the GAIN-ICAT2 study, but also from an institutional study, and then across institutions through something called the Genomic Evidence Neoplasia Information Exchange, or GENIE Consortium, which is supported by the AACR, to identify osteosarcoma biologic subtypes associated with poor response to upfront chemotherapy. And with that, I'd like to acknowledge a number of people, patients and families who participate in our studies, and our funders in particular, and of course, the people who do the work and the collaborators. Thank you so much.
All right, perfect. Thank you so much. And perhaps in the interest of time, let me just take one question so that perhaps you can hop on to the clinic. All right, so I think David Norris is asking, thinking in an adaptive sequential NF1 mode, in those cases where extraordinary responses did occur, how early did they become evident clinically?
Yeah, so I think you could see from the swimmer's plot that most of our partial responses occurred quite early in treatment. And I think that's actually primarily because these are kinase fusions. And I mean, this is still a little bit speculative, but I think in our clinical practice, most of us have seen that this is sort of the ideal scenario for the precision oncology approach, or for molecularly targeted therapy.
All right, outstanding. All right, so let's move on to the next speaker. Thank you so much, Dr. Janeway. I think that was a fantastic talk. I think pediatrics should be a priority, and thank you so much for putting everything into context and perspective. All right, go ahead, Mani. You want to introduce our next speaker?
Sure. Dr. Janeway, I know you have to go for your clinic. If you could answer the other questions posted online, even in your clinic when you get a few seconds, we would greatly appreciate it. Thanks again for the talk.
I will do that.
Thanks a million. Thank you. Thank you. And without further ado, we'll move on to the next presentation. And we have another wonderful presentation again, going to have a wonderful presentation again on pediatric oncology as well. We have our next speaker is Dr. Andrea Hayes Jarvin. She's a professor of pediatric surgery and surgical oncology at the University of North Carolina Children's Hospital. She's a Surgeon-in-Chief of the UNC Children's Hospital and the Division Chief of a Pediatric Surgery at UNC. Dr. Hayes has got a basic science lab as well as clinical research lab. In both, she's focusing on rare sarcomas, and in fact, she's mainly focusing on refractory and resistant tumors, particularly in children and in soft sarcomas. She was also at MD Anderson before she moved to North Carolina. She was a section chief here at MD Anderson Cancer Center. She's also an elected member of American Surgical Association, and she served as a chair of the various committees at American Pediatric Surgical Association as well. And in fact, she's in 2002, she became the first African American female pediatric surgeon board certified in the United States. And she performs a high-risk life-saving procedure in children with rare form of cancer and developed the first orthotopic xenograft model for metastatic Ewing sarcoma. Again, she's nationally, internationally recognized for her work, pioneering an operation known as HIPAC, or HIPAC for children. And thank you again, Dr. Jordan, for making time to give a presentation at this forum and educating our audience with your wonderful science and the clinical research. Thank you. Without further ado, I welcome Dr. Jordan.
Well, thank you all so much for having me. It's just wonderful to be invited to this great symposium. Okay. So today I'm going to talk about one of the diseases that I have a passion for that I've been studying for a number of years called desmoplastic small round cell tumor. And I'm using this platform to help us understand how we can learn from these rare diseases. And so today I'm hoping to demonstrate to you an approach to understanding this rare tumor using precision oncology and how my clinical observations in the operating room led to some laboratory questions and how that using precision oncology can be used to approach these rare heterogeneous tumors that are so difficult to study. So desmoplastic small round cell tumors is a very rare tumor that occurs in the abdominal cavity primarily. And we don't really know the exact incidence of it, but the SEER data seems to show that it's about 200 cases per year in the entire United States. So extremely rare cancer. And interestingly, it occurs in mostly male patients. So about 90% of the patients are male, and the median age is about 18. So it spans from the pediatric into the adult age group from age 5 to about age 30 on average. Those where most of the patients fall. It's interesting in that it's characterized by EWS-WT1 fusion that really defines this disease. So it's similar biologically to Ewing sarcoma in that it has the EWS and the WT1 for Wilms tumor. So it's interesting biologically because it's sort of like a fusion between those two very, very different clinical tumor types. And here's the splice variant that occurs between the EWS and the WT1 as you can see here. The main challenge with DSRCT is recurrence. So these patients, although they get initial chemotherapy and can have some response to their disease, overall they have a very poor survival. The tumor is also interesting because we don't really know where it starts. We know where we see it clinically once it presents. And most of the time it occurs in the abdomen. Sometimes it's extra-abdominal, and sometimes it's in both extra-abdominal regions and the liver, the lungs, lymph nodes, etc. You can see the most common locations of the tumor, but within the abdomen or in the liver and the lymph nodes and the rectovesical space and the rectosigmoid region. So the interesting thing about this tumor is we don't know where it starts. We don't know where it begins. It doesn't start with a typical one tumor lump like other tumors like breast cancer or lung cancer, where it starts with one tumor and then it can spread and be metastatic. All of the patients that have this disease present with multiple intra-abdominal tumors. I've not ever seen a patient in the couple hundred that I've seen that has had one tumor. So what is the organ of origin? Where does it start? Why is it so difficult to treat and has such a poor prognosis? And this is a histology that gives some clues to why this might be. So you see here these nests of small round blue cells, and this is an untreated DSRCT tumor. And you can also see the desmoplastic component of the tumor. And so the small round blue cells that are obviously undergoing quite a bit of mitosis or quite a bit of mitotic figures is a portion of the tumor that responds to chemotherapy for obvious reasons. So the cytotoxic sensitive tumors are these small round blue cell tumors that are rapidly dividing, whereas the desmoplastic component that has more quiescent cells and looks a bit more fibrotic and desmoplastic, hence the name, is not responsive to chemotherapy and is actually what remains at the time of surgery. So these patients undergo months of chemotherapy, and then they still have a large amount of disease burden within the abdominal cavities. I want to share with you some photos from inside the operating room so you can see how different this tumor is than other tumors. So in keeping this in mind, that the chemotherapy is going to reduce or shrink these rapidly dividing cells, what we're left with in the operating room is very fibrotic, white colored tumors that need a surgical resection. So this is looking up at the right diaphragm. My hand here is on the liver, and you can see the tumors here high that are stuck in the on the diaphragm surface itself. And you see very clearly that we're not talking about one tumor, we're talking about several hundreds of tumors, and this is just in one part of the abdominal cavity. This is another part of the abdominal cavity right on the surface of the small intestine. And you see these very tiny nodules. They're about 1 or 2 millimeters. This instrument is about 2.5 millimeters in thickness. And you can see very clearly that there are maybe 50 to 100, you can see just in this image, that are studding the surface of the small bowel. The other part of the abdomen that it's almost always presents in is the omentum. So this is a photo of the omentum in the operating room, and you see it's sort of like this net and web of fatty tissue with these round, hard white nodules of the treated tumor. These, of course, all these patients have been pretreated with chemotherapy. It occurred to me that when I operate on these patients, that almost all of them, 100% of them really have tumor in the omentum and plus minus the other locations. Now, the omentum sort of drapes over all of the bowel, so it's hard to know. Nobody knows too much about the omentum, but it's interesting to me in that it might be the organ of origin of this very rare tumor. So my hypothesis is that the omentum, it might be the necessary microenvironment necessary for the growth of this tumor. When you look up the literature on the omentum, it's only recently that we found some answers as to what it might be and what it might do. And I thought it was more than a coincidence that in this paper that describes cellular basis of omentum and using single cell RNA sequencing, they found that WT1 was a mesenchymal progenitor marker. And as I mentioned to you earlier, this tumor has a WT1 partner that defines its biology. And this WT1 is not only a marker for the omentum, but other types of mesoderm that are listed here. But most importantly, it could overlap with the DSRCT tumor expression. On the right here is some single cell sequencing from my laboratory that I'm going to go over in a little bit, but this is where you see the WT1 expression overlapping with the omentum markers. The other interesting thing about this tumor, remember I mentioned that 90% of the patients are male. Well, of course, that would bring to mind whether androgens were involved or whether the tumor cells were androgen sensitive. And here is when I very initially started looking at this when I was at MD Anderson. And I took DSRCT cells, for which, by the way, there's no commercial cell line. So these are cells from one of the partners. And this is a Ewing cell line, and this is a prostate cancer cell line. So you can see that the DSRCT cells are more closely mimic the prostate cancer than the Ewing's sarcoma cells. So this DSRCT, Ewing, and prostate cancer. And So what do we think the role is of this androgen receptor, as it's pretty clear that it looks more like a prostate cancer cell than it does a sarcoma cell? And can we capitalize on this when we're talking about novel therapies for DSRCT? We again look by immunohistochemistry to just confirm the same thing, that if you look at androgen receptor and the prostate cancer cell line, you see significant increased expression of androgen receptor in both the DSRCT and prostate cancer. When you add testosterone to the DSRCT cells, they again demonstrate sensitivity by increasing the proliferation by more than 50%. Here on the right side graph, you can see in red the prostate cancer cell line, in black the DSRCT, and in blue the Ewing sarcoma cell line. And when you add an inhibitor to testosterone, you can see that the red line, the prostate cancer cell line, most closely mimics the DSRCT cell line, and it's very separate from the sarcoma cell line for which it shares a genetic partner. So these overlapping pathways of prostate cancer make it seem like it's more similar to that disease. And so when you're developing a murine model, and the orthotopic xenograft model, as we were taught by Dr. Fiedler years ago and others, it's really critically important to understanding the disease. Of course, it's quite difficult to develop an orthotopic model if you don't know exactly the organ or tumor cell of origin. So the best guess we had based on this preliminary data was that the seminal vesicle area, the prostate cancer area, was where perhaps the tumor got its genesis. And so I developed this myself in my laboratory, we developed this orthotopic xenograft model of desmoplastic small round cell tumor, where the tumors we really inject around the seminal vesicles of the mouse. And when we did that, this peritesticular injection, of course, we used luciferase labeled DSRCT. And then in this particular model, it takes about 8 weeks for the tumors to grow and develop. We image them weekly, and then we apply treatment strategies to interrogate the model. And here's how it can look. This is a control mouse on the left. The second from the left is a mouse with 1 million cells, and then going over to 2 million cells in the far right. If you look on the right, and remember the images I showed you before in the operating room, this mouse looks very, very similar in its phenotype to the human patient. So this is the mouse seminal vesicles and testicular equivalent, and you can see these patches of white tumors that are here, here, and here, here, and then a small amount on the surface of the liver in 2 places. And so we really found that this injection site duplicated what we saw in the clinic. When you look microscopically, you also see microscopic metastases, primarily in the abdomen, as demonstrated by this peripancreatic tumor that's embedded next to the pancreas in the mouse, and then the supraclavicular lymph nodes, also in a very distant location. We were able to identify DSRCT. The immunohistochemistry can give us more clues about what these cell types are and what they come from. And so this is what I call the older way of looking at the cells, but can really give us some overview of what to look for when we drill down on a single cell level. So this again is the small round blue cell component that's within the desmoplastic cells. You see that it stains very brightly for WT1 in this portion of it. And it's, when you look at the Desmond and synaptospicin and the Omentum, it's very clear that this part of the tumor is expressing these markers. But when you look at ACTA, which is more of a stromal marker, you see that there's more highlights there and more staining there. Androgen receptor, interestingly, doesn't change too much before and after chemotherapy. So moving forward, what we wanted to do was drill down on the cells. What are the difference between these cells and these cells, between these cells and these cells? And so now we know we have precision oncology to use for that, and we can make some hypotheses. So again, this is now the Omentum. So this is our best guess as to where possibly the environmental stimulus for growth comes from. And when you look at the Omentum for the same proteins, you see that there's a little bit of WT1 and not really any synaptophysin or vimentin, but then some ACTA, which demonstrates some of the fibrous components of the Omentum. So using these DSRCT cells, we realize, as we do for every other tumor, that the cell lines are not going to be as specific as the actual tumor. So this is just to demonstrate that when we use the actual tumors from the patients, we get more accurate data than when we use the cell line. So what I started to do is get tissue from every single patient in the operating room, and having my laboratory technician take it immediately to the laboratory for cell dissociation and to do single cell sequencing. And the reason I believe this is more accurate, as you can see here in lane 4 and 5 are the DSRCT cell cDNA, whereas lane 3 is from the cell line, and they look very different. The human DSRCT tissue in lane 2 looks much more similar to itself than the cell line. So from here forward, we are using tumor cells directly from the operating room. So this single cell sequencing using RNA, this single cell sequencing is what we did is we dissociated cells from the fresh tissue and adjacent omentum of these patients. And the DSRCT patients we used to conduct a single cell sequencing, we also compared that to 2 normal omentum RNA-seq databases from 24 healthy individuals. And then we did some data filtering and some doublet removal and did unbiased clustering and cell type assignments, and showed that both expected and previously unrecognized cell populations within the tumor and omentum included several different cell types, which I'll show you shortly. We found that markers for each of these cell types and genes exclusively expressed in DSRCT, and we found a very small population of rare cancer stem cells. We had to filter out some of the adjacent omentum samples, so we used basically tumor with adjacent omentum, and also from adjacent omentum cells from patient number 1 and patient number 2. I'm just going to show you these 2 patients, which was our initial development of our single cell sequencing platform. So a single cell sequencing, as you know, varies in the specific technique depending on the tumor type. So we needed to develop this technique specifically for a sarcoma and DSRCT. So that took a bit of time to develop. And so here, just wanted to share with you how we can really use this precision oncology to drill down on exactly these tumor cells. So this is the WT1 portion of tumor cells. So these single cell populations, this is tumor 1, excuse me, patient number 1 and patient number 2. And these are the 2 major cell type populations identified by WT1. Remember, these are the small round blue cell component. This is the desmoplastic component. And you can see they're shared in the tumor cell populations. And this is to remind you of the immunohistochemistry for those cell types. And since none of the available reference databases had assigned markers, we had to assign those from mesothelial cells and what other cell populations we had in this tissue. And so over here on the far right is what we believe are the cancer stem cell populations that locate the WT1 and the ACTA2, which remember are the dividing cells and the stromal component. And we focused on those and highlighting. And we also want to make sure that they express androgen receptor based on our previous studies. So if you look here again in what we believe are the cancer stem cell population, they share this expression with the cell populations of the tumors and the first and second patient, but do not share those with the other cell clusters that represent mesothelial cells and some immune cells as well. This is, again, just a different way to look at it and assigning color to it. And you can focus on the fact that there are mainly the 2 cell populations in the cancer stem cells. SOX2 is known to be a stem cell protein and gene. And so when we see the genetic expression overlap with the SOX2 and the WT1 and the ACTA, we can identify this very small population of cells as likely being the cancer stem cells. Again, assigning different color to it, you can see them even in more detail on a cell-by-cell basis. The EbCam is another genetic stem cell population that we identified, again, within this small subset of cancer stem cells that are only scattered about in patient number 1 and patient number 2 cell clusters. So when we assign the clusters and we probe them, we can identify the different cell types. And you can see here that each cluster we've now assigned a different color to, these being the two different patients here. We've already talked about the stem cells component of it. And these other clusters represent, for example, these are neutrophils. These are the fibroblasts. The green cluster is actually the dead red blood cells. So it's a very specific, this advanced precision oncology. It lets us to really see into the single cell level to understand which expression each one of these cells represents. And these group of genes are now what we'll be focusing on that are in our future experiments with this cell group. And this is, again, another way to look at it. You can see here that there are neutrophils, NK cells, endothelial cells, macrophages, fibroblasts. And remember, these are just the dead red cells that fall out of the populations. Of course, in doing a heat map, you can verify that these cell populations that we've identified that are represented here across the top of the screen can be identified more specifically as oligodendrocytes, myofibroblasts, astrocytic cell components, mesenchymal stem cells, and endothelial cells, etc. We identified the populations and the clusters that we were interested in, that we hypothesized would drive the growth of this tumor. So this is WT1, this is androgen receptor, and these are the other genes that we believe are going to be important based on them overlapping in the populations with the WT1 and AR positive cells. So we're now drilling down on these genetic subtypes and the genes to understand more about this very complex tumor. Again, some more really very, very specific genomics is we started to look again at now we're just within the tumor cell population. So excluding the fibroblasts and the other cell populations, we wanted just to look at the tumor cells and the stem cells and these DSRCT patients. So then we can look at the percentage of each cell and each cell type between the treatment, the two tumor types, and it really helps us to be very specific about these components. And again, looking at the epithelial markers that might be present in the stem cell component, we've assigned the number cluster 10 in this particular case. And when you look at cluster 10, yeah, and the blue is one patient, the green is the other patient, it turns out that the patient in blue had a more chemo-resistant tumor and actually succumbed to their disease as opposed to the patient in green, which didn't. So this also allows you to look at the percentages of each cell type. So in this particular patient, they had more of that stem cells in their cell type than the other patient, than patient number 2, who did not and survived a longer period of time. The last thing I want to mention to you about how we can look specifically using genomics at these cells is when you can compare the tumor-like cells to the normal cells and then the adipocyte-like cells to the normal cells. Because our hypothesis is that the omentum is the organ of origin or the tissue of origin, you remember that the omentum is made up mostly of adipocytes or fat cells. So we want to know, what do these tumor cells look most like? Do they look most like the fibroblasts and the surrounding stroma? Do they look most like adipocytes? What do they mimic? And so we can do these comparisons to be able to see exactly what they mimic. And this is the transcriptional changes that we found in the fibroblast clusters. When I use the word fibroblast sort of lightly, as we identify them more specifically. And here is tumor versus normal versus adipocyte versus normal, and endo-like versus normal. And you can see the fold change here in the different genes that we've identified that are expressed in these different cell types. And the important thing about that is it allows us to get an idea of the proportion of what here assigned as the red and the black cells, but the comparison of the tumor versus adipocyte. And what we found was we saw the highest fold change when we were looking at the tumor compared to the adipocyte-like expression patterns. And so we again hypothesized that these adipocyte-like genes are more common to the tumor and therefore may be a clue as to this omentum being the organ of origin. The pseudotime analysis is another thing we can do to look at the burden of the tumor cells and really follow what looks like a tree or the RNA velocity, a ratio of unspliced to spliced variants as you go from what we believe are the stem cell populations, or actually you could look at it this way, from the tumor cell to the stem cell populations and the RNA velocity. So in conclusion, what we observed was that the DSRC T cells were identified as two subtypes, more of a myofibroblast subtype and one of the cancer stem cell marker subtypes. And the potential cancer stem cells and some immune cells and stroma cells were found exclusively in the tumor types compared to the omentum from normal patients without DSRCT. The RNA sequencing that we did using single cell sequencing really was done to identify this particular population of chemo-resistant cells and the overlap of genetic markers. And we found some marker genes that we're now going to be investigating, about 350 of these unique markers to determine which one we might use as a future drug target. So I want to thank you for your attention, and just thank you for the organizers for having me, and just wanted to just give you one example of how we can use very specific precision genomics to identify what we hope is going to be new treatment paths for the future. Thank you.
Fantastic, Dr. Hayes Jarvin. Thank you for the inspiring talk. Again, we call ourselves precision oncologists, but surgery is the ultimate precision oncology. It's the ultimate targeted therapy. You precisely, remove the tumor, even if there are zillion mutations. Thank you so much for showing and inspiring us on how focusing on an extremely rare unmet need tumor and how that can inform, just not just for that disease, but for several other diseases as well. Thank you so much. Mani, do you want to, we can take one question.
Yeah. No, I just have a question. So again, thank you. Again, being, you are able to translate finding for basic science to the lab, to the clinic. So the question I have is, you said the AR, androgen receptor, is expressed in these rare desmoplastic small cell round cancers, right, tumors. So is the AR expression is sex specific? In other words, do you see this in a male patient but not in a female patient, or is it independent of sex? Number one. If so, would you also expect to see, let's say if it is sex specific, then do you expect to see ER expression in female patients?
Yeah, that's a great question. Thank you for the question. It turns out that we see the AR expression whether they're male or female patients. The location of the tumors and how they present in the clinic, as far as having hundreds of abdominal tumors, is the same between both male and females. Interestingly, though, when I was first starting several years ago trying to develop this orthotopic xenograft model, we were using female mice sort of by accident, if you will, because usually we order female mice by default because they don't fight as much in the cages, et cetera. And then my postdoc pointed out, we're doing this in female mice, and that's why the tumors aren't growing. Well, once we changed to male mice, the tumors grow very well. So I think that the estrogen receptor part of it probably doesn't have much to do with it. And once we get more information about really the ontogeny of this tumor, we'll be able to answer some more of those questions.
Thank you. And also maybe isolating the stem cell population which you've shown through the single cell sequencing and characterize them. I'm sure you're kind of lining up those studies. It's wonderful talk. Thank you very much.
All right, thank you.
Thank you. Really appreciate your time.
So I think your voice is a little bit muffled. I don't know if you need to, yeah. I don't know if the other speakers hear that way. It was like,
okay.
I don't know. Ebony, do you hear?
I just messaged Dr. Mani too. It just seems a little muffled.
Okay, all right. I will fix it. I don't know why. I'm just using my laptop, MD Anderson laptop, and connected to my desktop.
Yeah, it's some robotic voice. All right.
I'll fix it.
While you fix that, I think let me go ahead.
I will. So I'll just introduce our next speaker, Vivek Sobeja. Dr. Vivek Sobeja is going to tell us what he's working on. I introduced him in the beginning, and he's a wonderful friend, colleague, and amazing scientist and a clinician. Without further ado, I'll invite Vivek to continue the presentation. Vivek, do you want to share your screen?
I'm sharing my screen now.
Oh, great. Wonderful. Thanks.
Can you see my screen? Let's see.
Not yet.
How about now? Can you see my screen?
Yes.
All right.
Perfect.
Perfect. All right. Thank you all for the joining this fantastic lineup of speakers. It's my pleasure and honor to talk about precision oncology, the next generation. These are my disclosures because of the nature of work I do in drug development. I will definitely discuss off-label use and our investigational use in my presentation. My agenda for today, I'll be talking about precision oncology, personalized medicine, and clinical translation and Fda approvals in biomarker-driven rare diseases. Also my brief experience with targets, specifically BRAF and why BRAF, and also recently the RET inhibitor, selective RET inhibitors. Again, how did we treat advanced and metastatic rare cancers before the genomic era, right? It was just like COVID. We treated cancers with anything that we know of, like any drug, anything that we use for malaria, for deworming, anything. And, moving on to the current era, what is personalized cancer medicine? The current practice is one treatment for the average patient. We do a trial and error method. And in personalized medicine, the right treatment is customized for the right patient at the right time with the right biology. Again, today is a historic moment when just minutes ago, we received full FDA approval of the Pfizer-BioNTech COVID vaccine. And this is going to be a household name. This is going to be called Comirnaty. It's going to be marketed as Comirnaty, which is a COVID vaccine. And this will definitely cascade off vaccine requirements by several institutions. So why COVID vaccine and cancer, right? Cancer has been impacted by COVID. Independently, COVID is a risk of mortality for patients with cancer. And there have been several studies published showing the determinants of mortality in patients with COVID-19 and cancer. COVID-19 vaccine is precision medicine. It's customized medicine, mRNA for the spike protein of the coronavirus. The vaccine triggers production of the spike protein and antibody specific to it. This is precision medicine. COVID-19 monoclonal antibody is precision medicine. So moving on back to oncology. So we have a lot of small molecule kinase inhibitors for multiple targets. And we have a host of immunotherapies starting from PD-1, PD-L1, CTLA-4, LAG-3, BTLA-4, ICOS, and so many hosts of immunotherapies coming up. So again, our work is to bring new drugs to patients with cancer, navigating the roadway of funding, space, staff, training, collaborating between industry, academia, FDA, NCI, all while providing multidisciplinary compassionate care to patients in the evolving landscape of regulations and also insurance issues. So in the next set of slides, I want to share, discuss about basket studies in oncology. And I don't want to go into the details because Dr. Drezner already spoke about basket studies in oncology. Talking to about my brief experience of the vemurafenib basket study and the ROR basket study, and also moving on, RET inhibitors in oncology. So why BRAF? BRAF V600 mutations are seen in 5 to 10% of human malignancies. Most of the tumors that express BRAF are rare or ultra-rare cancers, which leads to constitutive activation of the MAP kinase pathway. This is the most common BRAF mutation is the valine to glutamic acid substitution at codon 600. This is a driver mutation in several solid tumors, including melanoma, colorectal cancer, papillary thyroid cancer, lung cancer, among others, and also hematological malignancies like hairy cell leukemia and Erdheim-Chester disease. So we designed one of the first histology-independent basket studies that showed that vemurafenib can be a tissue-agnostic target. Initially, in non-small cell lung cancer, the response rate was 42%. And in Erdheim-Chester disease or Langhans histiocytosis, the response rate was 43%, which eventually led to the FDA approval. During the course of the study, we also found out that colorectal cancers with BRAF V600 alterations were unresponsive to monotherapy vemurafenib. And at the same time, there was a Nature paper published showing innate activation of the EGFR pathway as a resistance mechanism to BRAF V600 in colorectal cancer. So we combined the EGFR monoclonal antibody, cetuximab, as a part of the study. And we showed that we can elicit minimal responses in patients with a combination of cetuximab. And this established BRAF as a therapeutic target in multiple malignancies, with the exception of colorectal cancer. And even in colorectal cancer, responses can be elicited with the addition of EGFR monoclonal antibody. Again, this was, the whole basket study shows that among extremely rare cancers, including anaplastic thyroid cancers and tough to treat cancers like glioma and pancreatic cancer, BRAF V600E is an actionable gene. Science moved on. The MAP kinase pathway was shown to be a resistant pathway to BRAF inhibition, monotherapy inhibition. So in melanoma, the combination of BRAF and MEK was approved, which led to sustained inhibition. And we observed more prolonged and durable and anti-tumor effect in this space. This also delayed and prevented the potential development of resistance. In addition, with monotherapy BRAF, we see secondary malignancies and hyperproliferative skin lesions. And combination with MEK inhibitor could alter this effect. So we designed the ROR basket study, the rare oncology agnostic research. The study design was simple. Any patient who with rare cancers as listed, anaplastic thyroid cancer, biliary tract cancer, germ cell tumor, high-grade or low-grade glioma, as long as they harbor a BRAF V600E mutation, would be enrolled on this clinical trial and receive BRAF and MEK inhibitor. In this case, it was dabrafenib and trametinib until progressive disease or unacceptable toxicity. The primary endpoint was investigator-assessed objective response rate. As you know, the anaplastic thyroid cancer is one of the tough diseases known to man. It's one of the most lethal diseases known to man, and it is chemotherapy unresponsive. So the median survival is less than 6 months with anaplastic thyroid cancer. We found early in the course that around 25 to 40% of anaplastic thyroid cancer patients harbored BRAF V600E alteration. So when we enrolled these patients, we saw promising responses. In the initial 16 set patient cohort, we saw an objective response rate of 69%, which was never seen in this extremely lethal disease with any other treatment. And as you can see, the CT scans show dramatic responses in these patients. Based on a data set of less than 25 patients, the combination received US FDA approval. This was the first ever FDA approval in anaplastic thyroid cancer. As you can see, we saw some early success in precision oncology with vemurafenib in Erdheim-Chester disease and dabrafenib and trametinib in BRAF V600E positive anaplastic thyroid cancer. The road basket study continues on, and we recently published the dabrafenib and trametinib in patients with biliary tract cancer, showing an investigator-assessed objective response rate of 51%. Again, routine testing for BRAF V600E mutation should be considered for all patients with biliary tract cancer. And this is now included in the NCCN guidelines. Moving on to the next part of my talk, my recent experience with RET inhibitors. We'll be talking about RET aberrations in oncology, RET alterations in non-small cell lung cancer, and RET alterations in multiple other cancers. Talking briefly about multikinase drugs and also about selective RET inhibitors and the drug development pathway for selpercatinib and pralsetinib. So RET is a receptor tyrosine kinase required for normal development. Alterations in RET structure and function can lead to tumorigenesis. RET can be activated by two major mechanisms. One is activating point mutations and dimeric RET fusions. Dimeric RET fusions are seen most commonly in 1 to 2% of non-small cell lung cancer and 10 to 20% of papillary thyroid cancer. And activating point mutations are seen in 60% of sporadic medullary thyroid cancer or 100% of hereditary medullary thyroid cancer. And RET is one of the first oncogenic kinase fusions cloned from an epithelial tumor. It was identified in 1985. In 2012, this was identified in non-small cell lung cancer as a driver. And subsequently, it was identified as a driver oncogene in multiple malignancies. In 2017, we had the first selective RET inhibitors enter first-in-human clinical trials. As I said earlier, RET is activated by two major mechanisms in cancer, the RET fusions and RET mutations. A number of oncogenic RET fusions and activating point mutations have been identified to date. The most common fusion partner for non-small cell lung cancer is the KIF5B, and the most common RET mutation in medullary thyroid cancer is the C634 series and the M918 series. Again, why did we need a drug for RET cancer? As we all know, there are several small molecule kinase inhibitors like cabozantinib, vandatinib, lenvatinib, sorafenib, and multiple IBs that go after the RET pathway. But mainly, the activity of these small molecule kinase inhibitors are from ancillary RET activity. They have greater potency on several off-target kinases across the kinome, like the VEGFR2. This led to potential off-target toxicities like hypertension, hand-foot syndrome, proteinuria, thrombosis, and QT prolongation. So we needed superheroes for RET, selective RET inhibitors in cancer. So how do we define superheroes? So patients with RET-altered cancers before the advent of selective RET did not achieve the promise of precision therapy before 2020. So you can see most of the small molecule kinase inhibitors like vandatinib, cabozantinib were not as, the PFS was not as high as osimertinib, ceritinib, and crizotinib in lung cancer. So the objective response rate and PFS achieved with the multikinases in lung cancer are notably lower than those achieved with ALK, ROS1, EGFR mutated lung cancer. These are similar to those achieved with cytotoxic chemotherapy. Interestingly, patients with non-small cell lung cancer harboring RET fusions do not respond as well to immunotherapy as well. Let me share a case with you. This is a 49-year-old man with a history of advanced medullary thyroid cancer who received total thyroidectomy with RET M918T mutation. This patient presented with progressive disease after 6 multikinase inhibitor clinical studies over 7 years. The multikinase regimens included sorafenib, vandatinib, and cabozantinib. These two agents are FDA approved for the treatment of medullary thyroid cancer, and subsequently went on clinical trials with this investigational agent, MDCD516, and then RxDX105. And eventually he was enrolled in an investigator-initiated clinical trial with a multikinase inhibitor combining with an mTOR inhibitor. Each treatment was discontinued for disease progression in the liver. And most recently, this patient presented with hepatic hepatomegaly, large volume ascites, severe fatigue, and markedly decreased performance status. When this patient presented to my clinic, the patient was cachectic, had 30 bowel movements a day. His performance status was rapidly declining. Interestingly, the molecular analysis of the liquid biopsy of the circulating cell-free DNA isolated from the blood before enrolling on the vandetanib and everolimus clinical trial identified not just the founder mutation, but a resistance gatekeeper mutation. So we knew from preclinical data that this gatekeeper is a resistance mutation, and we did not have any agents clinically available to go after this resistance mutation. So what did we do next? So we knew from preclinical studies that this drug, LOXO-292, or selpercatinib, had high kinome selectivity for RET, and it was shown to overcome the gatekeeper alteration resistance in multiple models. In addition, it showed blood-brain barrier penetration as well. So this patient was treated with this agent called LOXO-292 or selpercatinib. A single patient, N of 1, intra-patient dose escalation study was designed using a compassionate use protocol. Again, as there was no human dose available for this drug, all the data was preclinical. We designed this N of 1 study and started at a low dose of 20 milligram once a day. And then we slowly intra-patient dose escalated from 20 to 60 to 80 to 120 to 160 milligram orally twice daily. There was resolution of diarrhea and pain, and his performance status rapidly improved even within the first week. The tumor markers, namely the calcitonin and CA, became normal. There was a reduction in tumor size by RECIST by 54%. Interestingly, as you can see, there was rapid, dramatic reduction of the circulating cell-free DNA. And at some point in the first 4 weeks after we initiated the single patient compassionate access use study in this patient, there was a bump. You can see that with the asterisks, that there was a bump in the tumor markers. This happened because patients could not come to Houston because we had a major 1,000-year flood event in Houston, the Hurricane Harvey. So he couldn't come to Houston to access drugs. The tumor was so sensitive, there was a rapid increase in the tumor markers. So once we could open the clinic, we opened the clinic and the patient could come back after 7 days and we could restart the patient, the tumor markers dramatically dropped down. So as you can see, this also shows sporadic mutation and the V804M gatekeeper patient response in the liver. Again, this reminded me how I felt on reading Chapman's Homer by John Keats. Then I felt like some watcher of the skies when a new planet swims into his ken, or like stout Cartus, when with eagle eyes, he stared at the Pacific and all his men looked at each other with wild surmise, silent upon a peak in Darien. So after this N of 1 case, the Libretto-01 clinical trial was going on in parallel. And in fact, the recommended phase 2 dose from the whole clinical trial was the same 160 milligram orally twice daily dose that was achieved in that N of 1 patient. And subsequently, this was published in the New England Journal of Medicine, and it received FDA approval. This waterfall plot shows the efficacy of selpercatinib in non-small cell lung cancer. And again, the overall response rate was 68%. And it showed responses regardless of prior receipt of chemotherapy, immune checkpoint therapy, or multi-kinase inhibitors. Again, this shows the efficacy of selpercatinib in treatment-naive patients. And based on this data of 144 patients in the primary analysis set consisting of 105 treatment refractory patients and 39 treatment-naive patients, the selpercatinib received FDA approval in lung cancer and medullary thyroid cancer and red fusion positive thyroid cancer. As Dr. Drezner mentioned, this was a line agnostic approval. The next set of slides, I'm going to share the story of Blue 667 or pralsetinib, also a selective, highly potent RET inhibitor. Pralsetinib was designed to treat RET-altered cancers. After showing high kinome selectivity for RET, it also showed more potent and selective than multi-kinases available at that time. As you can see, pralsetinib potently inhibits RET alterations and resistance mutants while sparing the VEGFR2. The ARROW first-in-human phase 1 study was designed using the Boeing design and with multiple dose expansion cohorts. The early phase 1 study showed dose-dependent exposure and RET pathway inhibition, showing MEK inhibition, ERK inhibition, dual phosphatase 6, and Sprouty4 mRNAs. Initially, the phase 1 trial also showed dose-dependent decline in MTC tumor markers, namely the carcinoembryonic antigen and calcitonin. And I got to tell the story of this young man. This was a patient, a young man, 27-year-old young adolescent young adult male, who presented to his local emergency center with a massive neck mass. He couldn't breathe. And there was a tracheostomy placed, and he was life flighted to our cancer center. Given the nature of the disease engulfing the carotid vessels, the trachea and the esophagus, at that time, only 2 FDA-approved drugs were cabozantinib and vandetanib was not an option, mainly because they targeted the VEGFR2. He would start bleeding from the major vessels. Radiation was also not an option given the risk of tracheoesophageal fistula formation. So we knew from the preclinical modeling that BLU-667 or pralsetinib currently demonstrated preclinical activity. So we started the patient at the phase 1 clinical trial at a dose of 60 milligrams. Slowly, we dose escalated to 100 milligrams, 190 milligrams, 200 milligrams. And the tumor markers started decreasing. And the patient had a fantastic response to therapy. The patient has an ongoing response to date with confirmed partial response of the tumors 3.5 years after the initiation of therapy. Again, he is able to do all his activities of daily living and is able to work with normal quality of life. 2 weeks ago, he called us that his child started kindergarten. Again, this is what makes an early phase clinical trial investigator wake up and go to the clinic to see such stories. Again, this was a similar story of a 37-year-old adolescent young adult mom with lung cancer who also had a dramatic response to pralsetinib or BLU-667. This is another patient, a 23-year-old woman with papillary thyroid cancer who presented several years ago with diffuse lung metastasis of papillary thyroid cancer. And she was always requiring supplemental oxygen right from diagnosis. She was treated with iodine-131 and had subsequent fibrosis. Her tumor progressed on sorafenib and then lenvatinib with increasing oxygen needs. Unfortunately, her performance status was rapidly declining as she was intubated 3 times over 6 weeks. At that time, because the BLU-667 or the pralsetinib clinical trial was available, we initiated, and the patient's tumor harbored a RET fusion, and we knew the activity. We started this patient at 400 milligram once daily, and patient had a dramatic turn of events. Within 4 to 5 months, the patient was weaned to Rumab. BMI, as you can see, the CT scans, the BMI steadily increased from 14 to 22.3 after 6 months. She remains on treatment after 3 years and started college, completed college, and is now searching for a job. So the ARROW trial since then continued and showed 73% objective response rate in treatment-naive RET fusion positive non-small cell lung cancer and 61% objective response rate in post-platinum patients. And again, in medullary thyroid cancer, the objective response rate in treatment-naive patients was 74%. And based on all this data, pralsetinib was also FDA approved in 2020 for the treatment of RET positive non-small cell lung cancer and RET positive thyroid cancers. Again, this was a line agnostic approval. Again, responses were observed regardless of treatment history, RET fusion partner, RET mutation, CNS involvement, and gatekeeper coverage. So see the timeline for FDA approval. In 2012, RET fusions were discovered in lung cancer. And 2017, we had not just 1, but 2 drugs enter clinical trials. And in 2020, we have both these drugs approved in this rare subset of tumors. Again, the 3 years from around approximately 3 years from first in human study. So, but tumors evolve. As you can see from this sketch from Charles Darwin, tumors recapitulate Darwin's theory of evolution. The original patient that I presented earlier, unfortunately, after 2.5 years, his tumors developed novel mutations beyond the gatekeeper mutation and the de novo mutation called the solvent front mutation and the hinge sites. Again, in our collaboration with Dr. Wu from Oklahoma University and this patient, we identified that resistance to selpercatinib and pralsetinib are found at the solvent front and hinge sites of the RET kinase domain. The identified resistant mutants are cross-resistant to both these agents because selpercatinib and pralsetinib use an unprecedented binding mode to dock into RET kinase. So we need a second generation RET inhibitor that cover the gatekeeper and the new solvent front mutations. That was on-target resistance mechanisms. This was an interesting case of a patient who developed an off-target resistance mechanism. Again, recapitulating the story of Charles Darwin's theory of evolution. In this case, an NTRK3 fusion was acquired as a resistance mechanism to selpercatinib in RET fusion positive non-small cell lung cancer. The light microscope invented in 1590 is still used to diagnose and classify cancer. We need universal genomic testing to win the war against cancer. Genomics is a diagnosis. Again, genomics just paints, one of the aspects of a complete molecular portrait. And we need, in the future, we definitely need something beyond genomics, metabolomics, transcriptomics, immunogenomics, microbiomics, and proteomics to have a complete molecular portrait of the patients in front of us. So the first time, as Dr. Drezner mentioned, the death rate from cancer declined by 29%, including a 2.2% drop from 2016 to 2017. The decline in deaths from lung cancer drove the record drop. Death fell for about 3%. One of the reasons why we are still analyzing is it could be from preventive measures and screening for lung cancer, a decrease in smoking. In addition, what we attribute this is to advent of immunotherapies and modern precision oncology targeted therapies. We had ALK, EGFR, and so many other host of targets in lung cancer. Again, we are all engulfed in COVID. Again, we thought we balanced the COVID with drugs and vaccines. Unfortunately, the Delta is posing a huge risk and challenge for us. We can counteract that by the recently approved COVID-19 vaccine, which is also precision medicine, and COVID-19 monoclonal antibody, which is also precision medicine. Again, this is the ultimate precision medicine that is working in day-to-day practice. The future, we are going to be seeing a lot more in precision oncology space with CRISPR-Cas9 engineering of T cells in cancer patients. Again, CRISPR technology and all hallmarks of cancer are being worked out. One of the silver linings of the pandemic is how clinical trials are going to be conducted. Again, Dr. Drezner mentioned about decentralization of clinical trials. Again, so far, all our clinical trials have been clinical trial centric. And we hope that the COVID pandemic and the cancer clinical trial pandemonium will find a silver lining from clinical trials moving on from being clinical trial centric to patient centric. In summary, this is my prediction. Explosion in drug development and costs are going to be different. It will change how early drug trials will be designed, will change where they will be conducted and who they will be conducted in, hopefully refractory patients. There's a revolution coming up in panomics is here and it's growing. Again, research biopsies, liquid biopsies, molecularly driven trials, independent of histology are going to be more and more. One of the most important things, and we have our amazing speaker, colleague and patient advocate, Dr. Ali Bricker, as from a patient perspective, talking about precision oncology. Patients will drive clinical research. There's going to be a lot more social media, search, and self-driven molecular testing. I'd like to thank all the patients and their motivated families and their motivated patients who enrolled on clinical trials, all the investigators and staff at MD Anderson Cancer Center and worldwide collaborating sites for the clinical trials. I'm always available on Twitter, and please feel free to email me for any questions. Thank you so much for patiently listening to me. Thank you so much.
Thanks, Vivek. That's a spectacular presentation. Thank you. I think
we have a couple of questions. Just one of them is, for me, is that for the students out there who wants to learn this bucket trial, maybe if you could explain what is a bucket trial, just in a nutshell. That way, just to get an understanding of regular traditional clinical trial versus bucket trial.
Oh, absolutely. So that was a great question. So there are two studies, right? One is the basket study. One is the umbrella study. Basket studies are histology agnostic, tumor agnostic studies, and patients are enrolled on the same, patients receive the same drug, and they are enrolled based on a common biomarker. As for instance, I mentioned, the BRAF V600E alteration does not matter if it's lung cancer, breast cancer, prostate cancer, glioblastoma. And, as long as the patient's tumor harbors a BRAF V600E alteration, the patient will be enrolled on this clinical trial. So that is a basket study. So histology independent, tissue agnostic, but the commonality is a biomarker. Again, that we don't focus on tissue of origin, we focus on the mutation. So that is a basket study. Umbrella study is different. So one tumor, multiple mutations. For instance, an umbrella study, the design of an umbrella study could be, a lung cancer patient walks into the clinic, I sequence a patient, the patient harbors a BRAF mutation, goes on a BRAF trial. Patient harbors a RET fusion, goes on a RET inhibitor trial. Patient harbors a HER2 alteration, goes on a HER2 trial, ALK, and so on and so forth. That is the umbrella trial. So umbrella trial is histology dependent. Basket study is histology independent. All right. Thank you so much, Dr. Mani. I think this is time. I think we
just one. There are a number of questions again asked in the chat, and it will be great if you could, again answer them on the side. There are, please try to post your questions on the Q&A rather than a chat. Because it's very hard to answer so that the answer could be visible to everybody. Again, thank you everyone for making time to join us to understand and then, about this precision oncology. We'll take a quick 10 minutes break. We are on time, even though we had some issues in the agenda. We are on time. We will meet at 10:30 Central Time, which will be 10 minutes from now, wherever you are, which part of the world you are in. Okay, see you in 10 minutes. Thank you.
Dr. Murray, I think once Dr. Mani joins in, we can go ahead.
And Suzanne is absolutely fine.
Wonderful. Thank you very much for, again, for all of you staying on and joining new. And so we will continue our this symposium with our next speaker, Dr. Suzanne Murray. She's a founder and the CEO of a company called XDAO Group International. She founded this in 1997, and she has been CEO of this company for the past 25 years. She has a lot of experience in organizational development, implementation, education, and behavioral research in health system performance improvement, etc. She supports various healthcare systems and industries, medical specialty societies and governments, both in Europe, United States, Canada, and Asia. She has spent 18 years at McGill University as a faculty in the Faculty of Medicine. She's also co-founder of the Center for Studies in Aging, and she was an administrator at the Montreal General Hospital. She has published very well and in various high-impact journals, and she's also a board member of Global Alliance for Medical Education, and as well as to the Journal of European CME. Thank you again, Suzanne, for agreeing to speak at this forum symposium. And without further ado, I pass the podium to you and thank you.
Thank you very much, Dr. Mani, and thank you, Dr. Sobeja, for inviting me for this particular session and very important symposia. Can everyone see my presentation in presentation mode?
Perfect. Yep. Perfect. Looks
excellent. So thank you. So again, just very quickly, I think Dr. Mani, thank you very much for a very nice introduction. And this particular session might be slightly different from the prior sessions because the focus will be on education and looking at a little bit of the science behind education, but also I will be focusing on disclosing a little bit about what the trends are internationally in relation to medical education and how it links and impacts precision medicine and oncology. Also, I'd like to just briefly, in 30 minutes, obviously we can't go through all the science of adult education, but I will give you some of those highlights. But more importantly, I'm sure many of you have experienced and are still experiencing the challenges around virtual education, either as a recipient of education or as an educator. So we will be looking at some of the best practices in a virtual world when it comes to education. But first, I'd like to start with asking you a question, and maybe you can type it in the chat. And if I can, invite maybe one or two of you to unmute yourselves, if I can have Ebony's support, to maybe share one or two comments in this regard. So I'd like you to think about for a moment, if you could name just one challenge that you are either facing or that you have been observing as when you are educating others or when you are attending education activities, especially since the pandemic in a virtual world. And I'd love to be able to get a sense of what maybe some of those challenges might be. So if you can take a moment just to type it in the chat, and would someone like to, if you don't know how to raise your hand, if Ebony, you can help out and maybe one or two comments on what those challenges might be. Who would like to share? And again, this is your learning time, so hopefully there will be someone that would like to share.
Okay, I'm watching. Let's see, Giselle. Unmute Giselle. I saw her first.
Great.
Giselle, you are now free to talk.
Hi. Great surprise. Thank you for having me. My name is Giselle Dutcher. I'm a oncology fellow and actually one of the chief fellows at the University of Miami, Jackson Memorial Hospital. So I'm currently a third-year fellow. So I started my training in 2019, just before the pandemic. And so as a result of the pandemic, most of our lectures went virtual for obvious reasons. And I think while we had some great opportunities to engage speakers from outside, it also led to, I would say, an overall loss in education as a whole, because it's just much harder to engage people virtually. In this current academic year, now as a chief fellow in a sort of educational role as well, we've tried to, or I've given some lectures already, and it's really hard to engage. It's really hard to not so much engage. You can engage people who want to be engaged with, but it's hard to assess the people who aren't engaging, their reasons why.
Okay.
Whether it's a me problem or a them problem or a situation problem.
Very good. Thank you. Yes, that is a challenge. And hopefully, as I'm speaking, I'll try to make sure that I address that further down. So I know we only have 30 minutes, but I see some very, very interesting comments in the chat as well. So the knowledge difference with the attendees, especially in a virtual context, and keeping oneself motivated in these horrible times, and motivated to learn, but also I'm sure motivated to educate when you can't see your audience, when you can't interact as much with your audience. So let's just take a quick look at the future of healthcare and education, and what we're seeing, not just in the future or what we're predicting for the future, but what we're seeing even currently, especially at an exponential speed given the pandemic. So we know that precision medicine, personalized medicine, and healthcare is absolutely critical, but there's digital tools, there's new approaches to practice and clinical decision-making, which is having a significant impact on specialists as well as other healthcare professionals. Also, the quantification of some of the environmental or lifestyle factors, and what data is valuable and what can be done with the data to have actually or yield an impact. So that's having what we call a cognitive impact on how this is affecting the learning ability of adults. And we're going to talk about that in a moment as well. Also, when we're referring to quality education now and where it's going, and it's going to continue to grow in this direction internationally, is going beyond knowledge transfer. And what we mean by that is that historically, in the past maybe 100 years, we've been focusing on knowledge, what we call knowledge dumping often, is where we transfer new studies, new guidelines. And that knowledge transfer, often in medicine, we've assumed that knowledge can be taken and then a clinician or a specialist can walk away and apply that in their context, when they're faced with complex patients or so forth, which is not necessarily yielding an actual impact on change in behavior or on clinical decision-making. So it goes beyond just knowledge transfer. And given we are in the virtual world, and how do you keep them engaged? I'm going to go through a little bit of that as well. So hopefully, as you said, that will answer one of your questions and some of you that posted some of those challenges in the chat. You're all familiar because you are all experts in personalized medicine, but just think for a moment all the changes in our health system and how we need to adapt education to those changes with the incorporation of personalized medicine. So even things like, diagnosing and selecting of tailored treatment, or the impact on your health system to ensure that there's the correct infrastructures for testing, whether it's biomarker testing, genetic testing, other forms as well, and also the impact on patients. So we need to take that into consideration. Telemedicine. So there's been a major rapid transition, as I'm sure many of you have experienced, in adapting from face-to-face to a virtual or a hybrid practice. And that has also has had an impact in relation to how we educate. And what's the ability of learning with all these massive transformational changes that are occurring, and the influx of a lot of rapid changing studies and treatments and changes in your health systems. So we're looking at education in a much different way. And that doesn't even talk about AI, the incorporation obviously into our practices and decision-making when it comes to artificial intelligence. And how it's changing the role of healthcare professionals as well. And that's going to continue very rapidly on a rise. Big data, real world data, and a lot of the data sets are often, we see it when we do international studies on, looking at healthcare professionals and specialists in oncology that are experiencing a lot of data coming from different sources. It's complex. They need to translate that data into clinical decisions, and that makes it much more complex than we saw historically. And the more traditional approach, obviously in oncology, especially when we're referring to community-based oncologists, looking at the differences between community-based oncologists, academic-based oncologists, or even other healthcare professionals. So making that data meaningful, knowing what's the right data, accurate data, all these things and phenomena are actually having an impact on the ability to learn, but more importantly, not just learning, but translating that learning into change in behavior, into performance in clinical settings. We're also seeing the impact of switching to value-based care. So focusing much more on what type of data will give us the more, I would say, reliable, valid information when it comes to outcomes, and how do we measure the outcomes, not just in relation to evidence-based medicine, but how do we measure and look at and evaluate the impact of the educational interventions that are being deployed now, especially virtually, which is much more challenging. So there are requirements, and those requirements are fundamental changes in education. We need to design based on not just evidence-based medicine, but evidence of what are the challenges, what are healthcare professionals faced with in all these new contexts and these new realities with precision medicine, new infrastructures and health systems and so forth, but also leadership. The leadership competencies that are required with care teams now that precision medicine is really coming into play much more in our health systems worldwide. And there's a shift. We're seeing a shift internationally from more specialty focused departments to multi-specialty collaboration, which also requires education that is going to be focusing a lot more on interprofessional teams, which is a different way of looking at education. So these are just some of the requirements that are needed. Now, I have another question for you, and I'd invite you to, again, write your responses in the chat, and I'll invite one person, if we can have a volunteer, to share. So what impact do you believe, you're in the context, you've experienced, I'm sure, many of these realities. What do you believe these changes are having on healthcare professionals or specialists like yourselves? Who would like to share? There's so many changes that have been happening since the pandemic. Anyone? Or in the chat? I can also invite Dr. Sobeja and Dr. Mani if they would like to share as well.
No, I think, from a healthcare perspective, Vivek could speak, but, from a science education point of view, it's very hard, when you organize a meeting in person, who are your audience, and therefore you kind of tailor your presentation to them. But here we have no idea, right? And everybody wants to learn, but not everyone is at the same level. Somebody may know in detail, and somebody may know, they just want to know what has been discussed. So it's so hard in these days, in this virtual day, to cover or address the expert to the naive patient, student or a trainee or a learner. So that's one of the challenge, at least I face, and we have been doing this meeting on a regular basis with Ebony's help, but it's so hard, and how to keep them attentive. But yes, they could address the question exactly what you asked for.
Well, thank you, Dr. Mani. And
the chat box, I think Akash Desai, one of the Mayo Clinic fellows, is asking the perils of overestimating one's ability to multitask on online activities, right, is challenging. And then another one, I think Sri Prasad is like keeping myself motivated in these horrible times and interact with people directly. Again, apparently he's been enrolled in a German university and had to do everything online from India. And another one is many oncologists are reluctant to, it's a challenge to educate the community in these times.
Absolutely. Absolutely. Thank you for sharing. So definitely the impact that we're seeing from a scientific perspective, given many studies that have been going on and published needs assessments in clinical journals, I might add, not just medical education peer-reviewed journals, is there are more complex patients. There's what we call a cognitive overload of science. that is really challenging. But if you add that to the multitasking. So as you're receiving science, as you're receiving new knowledge virtually, you're also looking at emails, you're thinking of the clinic. You're not in a congress anymore where you can actually focus. So the multitasking is something that is added to the inability to learn as efficiently as possible. And how do we address that? But in addition to that, as you might already be aware, many healthcare professionals and specialists are faced with being much more accountable for their competencies and their professional development. So many colleges, Royal Colleges, as well as, for example, the American Board of Medical Specialty Societies, have put forward competency models where the choice of medical education that a specialist chooses also has to be able to respond to some of these competencies that are now mandatory in many countries and by many licensing bodies or colleges. So this is really having a significant impact, and it's not just the virtual world, it's everything that also complements that. So we need to look at, as an educationalist, we need to look at new ways of working, new ways of designing education, educating educators, not train the trainers, but educating educators to facilitate learning. So I'll give you some tricks in a few moments before our time is up. And what we want to do is really become more familiar with the science of adult education. And just very quickly, historically, as I mentioned, we talked about CME, and in many countries, we still talk about CME, but the focus was really on knowledge acquisition or knowledge transfer. Over time, we saw an evolution that continuing health education was not just targeting or restricted to physicians, started to incorporate allied healthcare professionals as well as patients. Now we're talking about continuing professional development, which is making sure that our education, like the sessions today, are going to contribute to new knowledge, but also provide you with opportunities to develop skills, clinical decision-making skills. And those competencies would thereafter be transferred into performance improvement, PI, what we call PI, where we can actually see a change in behavior and measure a change in behavior in clinical performance. So how the actual education has impacted a change in behavior. Education is not an activity. It is a process. And the first I did it very well, some of you, is the needs assessment. Who's the audience? What are their challenges? Not necessarily their wants, but their challenges, their problems in their clinical setting. And the more you can conduct needs assessments at the beginning of a session or of a program or a curriculum, the better, because then you can tailor obviously the actual program to what we call behavioral objectives or learning objectives. And this is where your educational design comes later. Too frequently, we see the reverse happen. We see that, specialists are saying, we have a great topic, we found a great format, and we're going to design the program, not necessarily knowing who your audience is, but more importantly, what their challenges are, to adapt your content to that particular group of learners. And that's really something that we're hoping to share with you today, is it is a process. Education is not just an activity. And as I mentioned earlier, we are talking about evidence-based education, which means conducting needs assessments, assessing what the challenges are, and combining that with evidence-based medicine to impact clinical performance as well as professional development. I'm sure some of you have heard the expression, children are like sponges, and the science of pedagogy. So pedagogy is what we've been utilizing for several decades in medicine now, where it's very teacher-centric. And this is really the art and science of educating children, meaning that it is teacher-centric. The teacher makes the decisions, takes the decisions regarding the objectives, the content, the method, the pace and place of learning. And from this, why? Because children don't have prior knowledge or prior experience or have, different contexts. So they take everything in. That's why we say children are like sponges, and that's from the science of pedagogy. What we've seen in the last 8 to 10 years is the evolution of finally in the field of medicine, implementing and utilizing and referring to the science of andragogy. So if you leave this particular segment of this session remembering the science of andragogy, that will be a wonderful thing. It is the art and science of helping, supporting, and guiding adult learning, recognizing that adults have different learning needs and styles than children do, and that they have life experience, they have knowledge. You've been to med school, so you already have a wealth of knowledge, expertise, values, motivation. And that's why we don't talk anymore, or we shouldn't be talking anymore about teaching. We're talking about facilitating, because adult learning is much more learner-centric. And we want to make sure that the education can have a direct application, because adults like yourselves, like all of us, we want to be able to learn and apply, because we have obviously a lot of other obligations, multitasking you were referring to, that actually can impede. So we need to have knowledge that we can apply very quickly. Case-based learning is one instructional design that is used frequently and more in medicine. It's very helpful. Adult scaffold. We obviously have our prior knowledge and experience. Someone like Dr. Mani or Dr. Sobeja provides us with new knowledge, new studies, and then we're going to try and understand, okay, how do we fit this with what we've been doing for years, or what we know for the past several years? How do we now scaffold that into what we've already done to now change the way that we're actually practicing or making clinical decisions and so forth? So it's much more active versus passive. So very quickly, one thing I would hope that many of you will also walk away with today is understanding the difference between needs. So we're not talking about wants or desires. So often we'll say to organizers of various events, or even classroom, what are the wants or the desires of students or of colleagues? And those are not necessarily what you're trying to target when you want to have education that's going to be impactful. You want to target unperceived needs, which means what are the challenges, the problem. So it's problem-based education. We want to make sure that whatever we share as content will be able to address some challenges or clinical decision-making in different settings. It must be relevant for adults, and in med ed, it must be relevant. The content has to be relevant to the reality and the context of the targeted audience, targeted learners, and address those challenges in the workplace, in the real world setting. The other thing we often talk about is needs. So when we're talking again about needs, I mentioned earlier, not wants. What we're trying to really assess here when we're talking about needs, and that's why I was asking you questions at the very beginning, and you can do that virtually. You don't have to be face-to-face or do a full-blown study to assess the barriers and gaps. You can do it live. As an educator, you can ask a few questions. What are your challenges that you're facing? So this way, as educators, you can address some of that in your content as you're going through your content. But a need is really a gap. It's a gap in relation to what healthcare professionals know currently today, what they're doing today, versus what should be. What should they know and what should they be doing? And that's where the gaps are. And that's where the education should focus on, is really those gaps. And are they knowledge gaps? Are they skill gaps, resources, context? And when you put all of these together, that's when you can actually enhance and affect competencies of healthcare professionals. Because at the end of the day, what do we want our education to achieve? Well, historically, we would say satisfaction, a satisfaction survey at the very end of a session. Did they like the program, the speaker, the content, the setting? But really, that's not what we want. We want to know, did they learn something? But more importantly, were they able to change anything in their practice based on their learning? So there is a model that has been around since 2009 that is used in continuing medical education around the world. And it is the Moore-Green-Gallus model. Dr. Moore actually is at Vanderbilt University in the US. And there are different levels of measurement that are possible. So obviously we can't go through every single one today, but at least you'll become aware and stimulate some awareness that these models exist and how to measure the impact of good education, or maybe education that needs to be improved. So you have the levels of satisfaction, knowledge and skills, competencies and performance. And these things are measurable when you know the methodologies and the methods. And if you can't measure and see success, then how can you reward it? How can you reproduce it? How can you know that it's a time? Very quickly, I'd like to give you some little tricks of the trade when it comes to inspiring audiences, engaging learners, and translating it into practice, especially in our virtual world. So one thing that's maybe important to remember is that adults sometimes don't learn because they have their own ideas, they have their own knowledge and experience, obviously they also have their own agendas, and they're coming for something very specific, which means adults are very busy learning something other than what we may have in mind. And so we just have to keep that, which is different than pedagogy or just teaching or lecturing. We have to go beyond lecturing when we want to go beyond knowledge. So what does a good facilitator or educator need to do? If you don't want your audience to zone out, especially in a virtual world, in a virtual context, so here are some of the to-dos and not do's. One of the most important things cognitively, and sometimes it's not conscious, but adults, we as adults, we like to know what the goals are. What are we going to be learning in this session? And I think that this particular Dr. Manny and Dr. Sobeja did a wonderful job, and so did some of the presenters earlier today, did a very good job in sharing what the goals of their presentation would be. And some did have learning objectives. Adults really also want to know, is it going to be more passive? Is it lecturing? Will I have an opportunity to engage or not? And you want to make sure as an educator and a facilitator that you want to make sure your content is going beyond the classroom. And what we mean by that is translating how it can be utilized, what you're going to be presenting, how can it be utilized in the real world setting. The more you link it to the real world setting and not just knowledge, new knowledge, the more the learning is going to occur, the more probable the impact of actually changing what you're doing in practice. And at the end of the day, I'm sure as a successful educator, that's exactly what you'd like to do. Very quickly, asking or knowing who the audience is, you've already mentioned that, and asking what the goals are. You can use ARS systems and so forth. And in addition, you want to have the minimal amount of text, especially virtually. Virtually, it's difficult to stay engaged, as we mentioned earlier, the multitasking, emails, you're looking at emails, cameras on, off, and so forth. Images, pictures, some of the presenters today, Dr. Sobeja, excellent presentation. You had a lot of pictures and images, and that triggers much more learning capacity in a virtual context. And also the opportunity to engage, even if there's 200 people online, just inviting people to, ask a question like I did, write in the chat, invite one or two to share. When you're delivering, and I'm almost at the end, when you're delivering, thinking about, better not reading a text, more having a dialogue. And I think our hosts have done a wonderful job in doing that. Looking into the camera, too often we still see that there are people that are looking at a screen, looking at their slides. Imagine if I had spent 30 minutes looking like this. Think about that. So it's really important to do that. Also, we still see a lot of educators in oncology actually not looking into the camera, or the lighting is too dark, or their camera is like this. So can you imagine? If I had spent 30 minutes like this. So the tone, your voice tone, often it's very monotone. It's very important, even more important virtually, to keep your voice tone, dynamic, active as best as you can. Again, some of the presenters today did an excellent job in that. Overload of slides. Please don't contribute to the cognitive overload. What we mean by that, you have 10 minutes to present. We hear and we see 50 slides. it's impossible virtually from a learning perspective to really acquire all that information. So it's much better to be more selective. What are the key messages? What's the key information? And keep it focused and engage your audience, especially when it is virtual. Just keep in mind, as you're all going to be expert educators, hopefully after these 30 minutes, is that most professionals do not welcome the feeling of vulnerability or expose their lack of knowledge or skills or showcase any areas that need improving. So very, very important to ensure, as yourselves experts, to make people at ease, especially in a virtual context. And many are not comfortable with having their cameras on, as I'm sure you've experienced. But it's important because it is we're not used to seeing ourselves either. That takes up a lot of cognitive space. So just keep that in mind, that it is much more demanding to do education virtually. And I'm going to stop on this slide just to keep this in mind. If you have a disorganized clinician and you give them technology of any sort, you are going to have an even more disorganized clinician. So at the end of the day, when you think of everything that your audiences and yourselves, even as learners, need and require, just keep in mind that you got to break education down into bite-sized pieces. Less is better. Translating it into real world clinical decision making, especially in precision medicine, because it's not practice as usual. Food for thought. So I'm going to, at this point, I'd love to welcome any comments, feedback, questions. It could be in the Q&A or it could be live, as you wish. Any thoughts, comments, reactions, or questions?
Thank you. This is really wonderful. Some of you may be wondering, we've been talking about the precision oncology, but the topic is about education. So one of the things which Vivek and I talked about is that, it's very important that we need to educate what is precision oncology effectively, right? And that is where we thought, having a Suzanne would help us to see. Many of you who are in the audience are educators. So, we all, including myself, could use some of her methods to effectively communicate the precision oncology as well as other topics what we work on to our audience. So thank you, Suzanne, for that wonderful presentation. And any of you, any question? I think Vivek want to make a comment.
I think we have a lot of chat bar messages, wonderful thoughts, again, practical insights as you said. Again, one of the main important things that we see in precision oncology is, there was a recent publication showing that, more than half of the patients, even with lung cancer, which is a poster child of precision oncology, are not being sequenced, or not in NGS is not being done. And, again, for adult learners, again, who have been grandfathered into, practice, what would be the method of, training and dissemination of, practice changing studies? Again, especially, most of us as clinicians, when we go to major conferences, we have patient coverage, we have everything all taken care of here. And then when we move, we are able to focus on learning science and learning updates. So, but in the virtual world, I think we are multitasking. As I was conducting this conference, I was getting paged, I was getting called. Again, we are busy. Again, we can't delegate stuff because we know we are here, and we know. So, how can one learn? In this pandemic, and I'm sure there's going to be a lot more virtual meetings in the future. I think there's going to be a hybrid model. This is great. This is one of the silver linings of the pandemic, that this technology existed even before the pandemic. Everyone had Zoom and never knew how to even operate the Zoom camera until like March 14th or 15th. So, what would be our, advice to all in this regard? How do we, focus and exploit this, virtual learning?
Yes, well, that's an excellent question, Dr. Sobeja. We hear this across the globe. The most important thing to remember with adult learning, especially in a virtual context, is engagement. And that's why again, if there with Zoom, and there are other platforms as well, but most of the platforms now you can do breakout rooms. And the more there is interaction, the more you can have, for example, specific cases, whether it's patient cases or debating on guidelines, for example. The more you can engage the learners virtually, the less they'll be distracted in relation to other tasks. Because the adult learner is not a passive learner. The adult learner wants solutions. And the way that we adults think is we need to debate things out. We need to, have questions. That's why you have a lot of questions today. It's because we need to understand, we need to be able to and the more dialogue and interactivity, the better. And it is doable virtually. And the platforms can facilitate that with break. So it's just a matter of designing slightly differently than just having lecture after lecture and a traditional Q&A. There are much more innovative ways, considered innovative, but have been proven to be much more effective and keeping the learners engaged in a virtual context. I hope that answered your question.
There is one more question in the chat box. What can medical education learn from YouTube, a global free platform that operates with unseen public? There is no real-time interaction in YouTube, yet their massive success is undeniable. Thank you. How can, how can next gen, precision oncology and next generation sequencing and all that, how can we learn from disseminating just without knowing the audience?
Mm-hmm. Well, that's an excellent, YouTube is an excellent example, because what we see often is how the design, first of all, and how, even though it's not interactive, usually the speakers are pretty dynamic, usually, not always, but often it's a lot of new knowledge. So if it is new, then it's absolutely appropriate to do to lecture and utilize various platforms. It's when it's time to actually incorporate in the content, whether it's YouTube or any content, is to make sure that you give one or two examples of how to translate this into a context, into a certain clinical setting, or with a certain patient profile, or how to translate a certain guideline with certain patient profiles. And precision medicine has a lot of new content because it is new. So it's okay to have some lectures. But when we're a little further down the road and you want them to translate that new knowledge into their practice, then you have to incorporate more case-based learning from an instructional design perspective, even in a virtual context. Debates. Often debates are perceived as negative, but it's the best way that adults learn. And we've just started implementing this in medicine in maybe the last decade or so, but it has been utilized in other industries and contexts, and from a scientific perspective in many settings. So just again, YouTube is very effective when it's new knowledge, very effective.
Perfect. Thank you so much, Dr. Moray. So again, given that our time is tight, we will move on to the next speaker. So it's my pleasure and honor to introduce Dr. Ali Obricker, neuroradiologist, physician, clinician, scientist, patient advocate, inspiring mom. And, most importantly, again, she, I know Ali really well. We did internship together again that was maybe 10, 15 years ago. I don't even remember, in Cleveland, Ohio. So she was born and raised in east side suburbs of Cleveland, Ohio. And she earned her bachelor's degree and her medical degree from Case Western Reserve University in 2002 and 2006. And we crossed paths when she did her internship in the Case Western Reserve University Metro Health Hospital. And she went on to complete her residency in training in radiology at the Cleveland Clinic, where she remained for a neuroradiology fellowship. In late 2015, after delivering her second daughter, Dr. Bricker was diagnosed with de novo stage 4 non-small cell lung cancer with brain and bone metastasis. As a 34-year-old mom, never smoker, this diagnosis came as a complete shock. Although genetic testing revealed a red alteration, her lung cancer, there were no targeted therapies available at this time. Now, 5+ years after diagnosis, Dr. Bricker and a handful of other RET-positive cancer patients and caregivers have founded the redpositive.org to promote awareness and fund research aimed at improving quality of life and life expectancy for RET positive patients. Through their partnership with Longevity, the RET positive endeavors to raise funds specifically to advance medical research targeting RET-driven cancers and precision oncology. From a young mother, a full-time physician, to a stage 4 cancer patient, and now RET positive cancer advocate, Dr. Bricker has experienced personally the life-saving power of precision oncology firsthand. Her daughters are now 5 and 10, and she is grateful for it every day. We are grateful for you, Dr. Bricker, to make the time and talk to us. Thank you so much. You've been such an inspiration, for me personally and professionally to pursue precision oncology. Thank you so much.
Thank you, Vivek. Wow, what an introduction. Thank you so much. So, I wish that I had received that lecture about sort of educating in the Zoom era before I put this together, but hopefully let's see if I can do a reasonable job. Let me share my screen. And please let me know if you can see this. All right, are you able to hear me and see my screen?
Absolutely, absolutely.
Okay, great. I think I can go in to this presentation mode. All right, here we go. So, hello, my name's Aaliyah Bricker, Dr. Aaliyah Bricker, and Vivek and Dr. Mani invited me here today to really just talk about the tremendous impact that precision oncology has had on my life. So, before my diagnosis, I was really not too different from probably many of the young medical professionals in the audience today. Before my diagnosis, I was a full-time physician. I was a young mom. I had just finished my 5-year radiology residency. As Vivek said, we overlapped in our intern year in the trenches in Cleveland. And I had finished a 2-year fellowship in neuroradiology at the clinic. And of course, as many know, the prime years of starting a family often overlap with the most intensive years of training in medicine. So I had just started a family. But I was excited to have recently joined the staff, finally in attending, life was good. So in my 2nd year and then attending, and while pregnant with our 2nd child, I developed a dry cough. I immediately dismissed this, as probably many busy professionals and young people do, as probably some viral respiratory infection or who knows what. My oldest was 4, and I had also developed a little bit of intermittent back pain, but I was so pregnant. This picture is from August when we went to Niagara Falls, that which tremendously pregnant ladies who are on their feet all day, I think a little bit of back pain is not that unusual, especially given that I was still working full time, and during fluoro guided procedures, I was actually wearing double the layer of lead to protect the unborn baby. So, these symptoms were really vague and easy to dismiss. I had never smoked, so the thought of lung cancer literally never crossed my mind. And of course, in retrospect, how naive I was to think that only smokers are at risk for lung cancer. So in my case, unfortunately, these relatively vague and mundane symptoms portended a much darker diagnosis. The nurses on labor and delivery were quickly alerted, that my pulse ox was a little low, in the low 90s, so not quite normal for an otherwise healthy 34-year-old. And soon enough, after delivering my youngest, an X-ray revealed a lung mass with sizable effusions, bilateral pulmonary nodules. And days after delivering my youngest, I was diagnosed with de novo stage 4 lung. So by the time I received this diagnosis, I already had a solitary brain metastasis and pretty extensive bone mets throughout my spine. I think, just to give a little bit of perspective, it's really difficult to describe just how devastating this diagnosis was for my little family. so many carefully laid plans for the future, starting a family, building a home, finishing your medical training. It all came tumbling down in an instant. And at the time in 2015, the notoriety of some of the ALK and EGFR inhibitors had started to grow. But when the routine biomarker testing at my home institution came back negative for ALK, EGFR, and ROS1, this was yet another heartbreak. With no actionable biomarker and already widespread disease, my prognosis was not good, probably around 6 to 12 months. So it was really at this low point in my story that things took a somewhat unexpected turn. And believe it or not, social media gets a lot of bad reputation, but my first stroke of good luck came thanks to the power of social media. Having originally posted the news of my youngest daughter's birth on Facebook, like literally the day she was born, I posted a picture and, we're so happy to welcome her. So in these days that followed after coming home with such a grave diagnosis, I was getting bombarded with all these well-meaning congratulatory messages from friends near and far. And it quickly just became unbearable. So many people congratulating me on my daughter's birth when I wasn't even sure that I'd live long enough for her to remember me. Finally, rather than recount my traumatic news over and over again to each person individually, I just decided to share the whole diagnosis story, everything, very publicly on Facebook. And this was kind of a bold step. Admittedly, not everybody feels up to doing this, but for me, I was too many congratulatory messages. And I'm glad that I did, because it was actually Dr. Sobeja who happened to see this post. After our intern year at the same Cleveland hospital, we both went off to our respective subspecialty training in different cities, but thankfully we were friends on Facebook. So Dr. Sobeja, took the time to write me directly and basically just urged me not to be put down by the initial screening results and to really get my tumor sample sent for more extensive genetic testing, which at the time, it was not normal to do this and it cost a lot of money, but obviously worth every penny. So at the time, this meant sending my effusion sample for Foundation Medicine sequencing. And I'm so thankful that he did this, because it was in these early weeks that we found out my lung cancer was driven by a RET fusion. So, although RET was not yet targetable back in 2015, when I was diagnosed, just over the phone, Dr. Sobeja sort of told me about some promising preclinical work that was underway, demonstrating that, in fact, RET-driven cancer could possibly someday, most likely, be inhibited just as potently and perhaps just as durably as the other cool kid driver mutations like the ALKs or ROS or EGFRs. And really, when you received bad news after bad news and your whole world has basically imploded, hearing this was like a ray of hope. And the power of that is really, difficult to put into words. So with this in the back pocket, in the back of my mind, this idea that maybe someday RET could be inhibited, I started my maternity leave with gamma knife radiation to my solitary brain metastasis. Here are the Band-Aids from the head frame. I started chemo right away. I had a Pleurex catheter placed for recurrent effusions, and I just dove right into platinum doublet chemotherapy. And literally my singular goal was to stay alive long enough to see maybe one of these RET inhibitors in action. And luckily for me, I had a pretty fair response to chemo. I was able to continue pemetrexed or Alimta maintenance for the next year and a half. And by this time, I'd already met several other ALK and EGFR survivors who were doing really well and made survivorship look really smooth. So I have to say, during that first year and a half, we really squeezed the marrow out of that year and a half, even though life on chemo is not that easy. So generally 3-week infusions, but the first week out of every 3, you're just crushing fatigue and sort of insurmountable nausea. But still, we went to Yosemite, we celebrated Halloween, we squeezed in a Caribbean vacation. I think, we did pretty well. Unfortunately, though, by summer of 2017, I began to progress again. And probably as many of those in the audience, and especially the panelists, have seen, when some of these cancers that are oncogene driven, when they begin to progress, it can be a very slippery slope. We were trying immunotherapy, which we now know does not really work in RET-driven malignancies, so it did not work in me. I tried a multi-kinase inhibitor, which for a second, maybe we thought was keeping me stable, but alas, it did not. Here, in fact, you can see the cabozantinib turned my hair platinum white, like one of the X-Men characters from those movies. But if it had worked, that would have been super cool, but it did not work. And then I tried second-line chemo, docetaxel plus carboplatin, lost all my hair. And to rapidly fail from one therapy to the next was completely demoralizing. So by late summer, I had developed new liver mets. I had actually, strangely enough, developed dermal mets, which grew along the tract of that Pleurex catheter, which had been removed. And perhaps the worst part was that I was completely oxygen dependent, even at rest. So, at home, we have a gas range stove, and it's one thing to be tethered to your home, tethered to one floor, no less. And I couldn't cook, so the whole family was suffering. Anyways, we were not in a good place. And this is when my story took yet another fortuitous turn. It's funny that Dr. Sobeja showed some of the RET inhibitors and the other oncogene, TKIs as superheroes. And that's literally quite valid in my story. So, just as I was getting washed out to sea and really had no other options left, finally, one of those clinical trials that I had been waiting for since 2015 for a first of its kind RET inhibitor opened at my home institution. So in September 2017, I was the first at my institution, the Cleveland Clinic, to enroll in the LOXO 292 trial. It was still in phase 1A dose escalation. And I think I was, among the first dozen or so humans to try this drug. So now this drug has been approved as Percatinib or Retevmo. By Halloween, just 1 month after starting Percatinib or Retevmo, I experienced a Lazarus-like effect. So the searing pain from my dermal mets, which I had never imagined would be so painful, it disappeared immediately. I wasn't even started on the effective dose. I was started 40 milligrams twice a day, but the pain disappeared. Within days, the visible skin lesions faded. By my first set of scans at 6 weeks, my liver mets had completely disappeared, and my pulmonary disease improved so dramatically that I was oxygen-free. Not only was I oxygen-free, but I was able to go trick-or-treating with my 2 young daughters. Being able to sleep without oxygen, get out of my house, it felt like a miracle. We even bought a Peloton bike and I was able to start exercising again, which is another ingredient to feeling human. So this RET-targeted therapy, it gave me my life back. Food tasted delicious again. I was exercising out and about. And although there is a percentage, I shouldn't over fantasize being on a targeted therapy, there is a percentage who cannot tolerate the RET TKIs because of transaminitis. But really for me, the only side effect I experienced was dry mouth. This was a far cry from the cyclic week-long nausea and fatigue that I had grown accustomed to during chemo. So that first year, we were all so ecstatic that I even convinced my husband to sort of wear these absurd Christmas pajamas for a coordinated Christmas morning photo shoot, which, you know, hasn't happened since then, but that's okay. We were all very happy. And I, you know, despite even having lived this experience, I still find it difficult to conceptualize just what a superstar this targeted therapy was for me. I mean, even though I was started at just 1/4 the eventual effective dose of selpercatinib or Retevmo back when I first enrolled, this single agent kept me stable for nearly 4 years. And this was not 4 years of sort of barely squeaking out an existence and, you know, walking around mostly like a ghost. This was living with capital letters. We squeezed in several Caribbean vacations. We went to visit Big Sur, saw the redwood trees. 2019, we braved the altitude of Breckenridge, Colorado, which is not something most stage 4 lung cancer patients who are formerly oxygen dependent would be able to do. But here I am skiing with my girls. We started a vegetable garden. We went to Costa Rica. I mean, it was wonderful. And then of course, during the pandemic, full of bike rides, more skiing, hiking in the woods. I mean, within the first 1.5 years of living with stage 4 metastatic adenocarcinoma, I had progressed through 4 different treatments. And yet here I was, a single targeted therapy, specifically zeroing in on my cancer's driver mutation, kept me living my best life for 4 years. And I wasn't even started at full dose. So the impact of these drugs and the impact of precision oncology is tremendous. Once selpercatinib and another second RET inhibitor, pralsetinib, became FDA approved, more and more RET survivors began to emerge. On social media, we began to find each other. So I went from knowing not a single other person with the same oncogene driver at diagnosis to being part of a RET-specific Facebook group with around 200 members and growing worldwide. It seems to be the next natural step in this evolution. But once we found each other, myself and a few other survivors founded an advocacy group, RET Positive, specifically to create a sense of community, raise awareness, and to fundraise for RET oncogene-specific medical research. So this is really the perfect segue to just mention that RET Positive, in collaboration with Longevity and the Hamui Foundation, just recently announced a RET-focused research funding award for up to $1.2 million. So if there are any interested applicants or bright young minds in the audience, applications and letters of intent are due September 17th. So I welcome anybody interested to apply. And it's really, again, a testament to how precision oncology has transformed so many people's lives, that we as survivors and patients are feeling well enough and living long enough and feeling empowered enough to even form these advocacy groups at all. So these groups based solely on the oncogenic driver, there are many of them now, and you can see from this timeline, the EGFRs and the ALKs, of course, sort of led the way in ROS1. But now there are several of these groups, BRAF bombers, the NTRKers, exon 20, the EGFR resistors, MET crusaders, and for RET positive. And, you know, these groups fundraise, and we've begun to mobilize the global community to drive the research, and it really feels like the sky is the limit. So in conclusion, if someone had told me back at Diagnosis in 2015 that I would still be here over 5.5 years, really almost 6 years later, it'll be 6 years in a couple months, I would have laughed in their face. And yet here I am, my newborn, here she was a newborn at Diagnosis, Now she turned 6 in November, and she's starting kindergarten tomorrow. And my oldest is turning into a sassy preteen. So I'm so lucky to be here to see this. Finding that my cancer was driven by RET and then actually living long enough to go on a drug that effectively targeted my oncogene driver has, again, it's been a total game changer. And what's even more miraculous to me is that each week in our online groups, we meet more and more people with not just lung or thyroid RET-driven malignancy, but neuroendocrine, anaplastic thyroid, colon, and just this past week, someone with squamous cell carcinoma of the head and neck that is positive for RET. These people are all seemingly deriving benefit from RET targeted therapies, which is incredible. It's transformative. So although eventually every malignancy placed under the pressure of a TKI or a targeted therapy will probably develop resistance, my sincere hope is, through continued interest and research into each of these driver oncogenes, hopefully we can usher in a new era where these oncogene-driven cancers no longer represent an existential threat, but instead a chronic illness that we can effectively manage with targeted therapies. I think my story, I just wanted to share to show everybody a glimpse of day-to-day living with this and how powerful precision oncology really is. So that's all.
Thank you so much, Ali, for the inspiring story. Again, we have so many messages from the chat, and I can read some. This gives me goosebumps. Erica Hansen Brown says it gives her goosebumps. Yes, of course, it gave me goosebumps, and it's hard to fight tears and control it, of course. So, and as someone who worked on the Percatinib trial and underwent an FDA inspection for the MDA, this presentation has truly humanized the efforts.
Vivek, we can, your voice is a little bit low. If you could, no, this is, really, it's mind blowing what you've gone through. And, we wish our best that this medicine works like a charm for you for the rest of your life, hundreds until 100 or more. And thank you again for sharing. Vivek, go ahead if you.
Yeah, thank you so much. Can you hear me better now?
I don't know about others, but it's a little low.
Can you hear me?
Yeah, go ahead. I think probably we can hear you, but not like loud.
All right. So again, this is powerful. This is powerful. Thank you for the truly inspiring story. And again, as someone said, Erica Hansen Brown said, it gives her goosebumps. And again, there's so many people working on RET here are all inspired. And I think Dr. Jerry Wu from Oklahoma University also says thank you. And his lab is been doing RET work for a long time. And they have discovered several mechanisms of resistance, which we are trying to replicate. What we discovered in the clinic, he's trying to help in the lab. So this is truly inspiring. I think this is a huge team effort, and this is a powerful story. Thank you so much. And this is what makes us all come together and work against cancer. One cancer at a time, one gene at a time, and we can do it. Thank you so much.
My pleasure.
No, thank you. It's not easy to come out, like as you said, into the Facebook, just posting, and then there comes Vivek Sobeja, and there comes RET, and here we are. And many times we all kind of worry about social media, but at the same time, as you said, it's making an impact. So thank you again. And I think there are a number of comments you could see in the chat as well as in the, so.
Sure, thank you.
It's inspiring and moving. Thank you. And thank you for this again, for the wonderful talk. And then I think, I guess there are no questions there. Yeah, everybody's kind of inspired, motivated, and understood the purpose of doing research, understood the purpose of doing a clinical trial, a basket clinical trial and whatnot. Thank you. So with that, we will move on to our next speaker, Dr. Ray Kurzrock. And, I'm sure many of you know Ray. Ray, when she was at the MD Anderson, for example, she started doing this precision targeting with, at that time, very kind of primitive technology. But, she has continued to remain on the forefront of this area to transform the medical care through precision oncology. And she's the chief medical officer of WIN Consortium. She's a world-renowned physician scientist, and she focused on development of novel therapeutics in oncology. She was the founding member or person who developed and also shared the first, one of the largest phase 1 clinical trial department at MD Anderson. And then she promoted and she has been promoting precision medicine. She's also one of the pioneer of the clinical trial called Winter Precision Medical Trial, focusing on the first time on transcriptomics and other genomics. This trial was the signature study of the WIN International Consortium, for which now she's the chief medical officer. And when she was at UCSD, she was the one who designed the personalized cancer therapy, as well as the experimental therapeutics program at the UCSD. And she also founded a rare tumor clinic focused on precision medicine. Her signature study of the center was the iPredict study, which is one of the signature study that gave for the first time individualized matched combination of treatments to patients with lethal malignancies, and then resulting in improved outcomes. She has published extensively, and she has been a major voice in this precision oncology space, and she's a wonderful friend, colleague, and a scientist. Thank you, Ray, for making time to give a presentation at this meeting. And without further ado, I pass the podium to you, and looking forward to learn. Thank you.
Well, can you hear me?
Yes, we can.
Yes.
Well, thank you for that beautiful introduction, and really want to thank the people before me, Dr. Bricker for sharing, and Dr. Sobeja for his work and the invitation. And I'm going to share screen, and hopefully it'll turn out. So give me a second here to get my screen. So can everybody see that?
Yes.
Okay, great. I'm going to talk about the future of precision medicine, the next steps, and some of what we've been doing for the next steps. And I'm always fascinated by analogies. And so that I put the picture of the brain cell and the universe on here, because it's kind of fascinating how they look similar. At any rate, these are my disclosures. And then I wanted to talk a little bit, just expand on the background. So most recently, I was at University of California, San Diego. I had several hats, but the most important one, the closest to my heart, was the Center for Personalized Cancer Therapy that I founded and directed. And before that, I was, as mentioned, as MD Anderson Cancer Center. I was the founding chair of Investigational Cancer Therapeutics, which is the phase 1 clinical trials program. Dr. Sobeja was probably my most famous hire. So he's been a phenomenal success in bringing the field forward. And really, a lot of my thinking about precision medicine started at MD Anderson and was informed by the work at MD Anderson, and then was expanded on at University of California. So I want to give you the take-home points from this talk. It's about giving the right drug, and it's really the right drugs to the right patient at the right time. At the genomic level, every metastatic tumor is unique and complicated. And so I've been living in Houston and San Diego for a long period of time. They're obviously very warm cities, but I come from Canada. And so I know a little bit about snowflakes. And I think metastatic tumors remind me of snowflakes. Each snowflake is different in crystal structure from every other snowflake. And every snowflake is unique. And it's very complicated. And so that I'm going to talk a little bit about that, because that's really the next generation of precision medicine. It is wonderful when we get responses to single agents like RET. But for most patients, their tumors are too complicated, especially if they're metastatic tumors, and they need more than one agent. I think that's really the next generation of studies that we've been doing that I think really needs to come globally. So metastatic disease requires customized, individualized combination therapy for the most part, not single agents. The pillars of precision medicine are genomics and immunotherapy. And they are not separate, they're married to each other. And finally, immunotherapy is best suited for really complex disease and in ways that, again, were unimaginable just a few years ago. So what has driven what I think is a complete transformation in oncology is really technology. And I remember in the 1990s when the conventional wisdom was that the human genome would never be sequenced, never. But never was over by about 2003 and when the first human genome was sequenced. And unfortunately, it took a long time, though. It took about 13 years to sequence a single genome. Today, we can do genomic sequencing in a day. It cost $2.7 billion to sequence the first human genome. By about 2007, we were down to about $100 million, and that's still pretty expensive for insurance per patient or for my lab. But today we can get a sequence for about $1,000. So when people say it's expensive, sure, it's expensive, but the drop in price and what has happened here is really breathtaking progress that I think is unparalleled in human history. So this has led to new information, which has led to evolution of clinical trial design. And we started out not too long ago thinking this is just an example that lung cancer was one disease. And today we understand that lung cancer is many different subsets of disease defined by their molecular alterations, maybe RET, maybe EGFR, HER2, ALK, and so forth. And then we went one more step, and we began to realize that the same molecular alteration might drive different cancers, and that we could target the alteration in different cancers. And it was really the genomics that was driving these cancers, not the organ of origin. And so that led to a whole new type of trial, whereas the trials that were the gold standard for most of my lifetime as a physician were large randomized trials with unselected patients. Very few patients would respond, and you really needed the randomized trial to see which one arm was better. But now we've moved to these small, highly selected trials where actually in many of the trials, we're picking the patients by biomarker, and most of the patients respond, or at least half of the patients may respond. And this is a totally new model. But this is really stage one of redesigning cancer trials. And it's really what's happened over the last 15 years when we started doing some of these histology agnostic trials at MD Anderson circa 2007. People literally thought we were a bit crazy, but now it's accepted and there's also FDA approvals. And what I'm going to try and bring you as we go along the talk is to stage two, which is just emerging on the scene. And it's the next stage, and it's where every patient gets a different therapy that is customized to that patient. And that's a whole new paradigm, but that's what the science is telling us to do. So the stage one gave birth to a new era. And again, almost unimaginable 15 years ago, tissue agnostic approvals driven by genomics. And so the first one was anti-PD-1 checkpoint inhibitor, pembrolizumab for solid tumors based on mismatch repair gene defect. And there were three things that make this date of May 23rd, 2017, when this approval happened, really remarkable. And the first two most people know about. The third one, a lot of people don't know about, and may be the most important aspect. So it was the first tissue agnostic approval. The approval was for all solid tumors, didn't matter if you had colon or lung cancer, or breast cancer. The second part was that the approval was based on a genomic marker, and that was mismatch repair gene defect or microsatellite instability. And the third part, which I think is really maybe the most important part, that the approval was based on retrospective real world data. And I think that's the least known out of it, but it opened the doors to using the computerized databases that we now have for approvals. And this was certainly not the last one. There's other drugs like palbociclib for male breast cancer that have been now fully approved on real world data. So this is still controversial, but to me, it's an extraordinarily positive happening, because it means that you don't even have to do a prospective trial in some situations if the data is already there. And that can speed things for cancer patients. And cancer patients need speed. They can't wait, as you heard from the talk before, you can't wait 5 years. They're not going to be alive 5 years to wait for new approvals. So pembrolizumab was the start, but it wasn't, certainly wasn't the end. Larotrectinib is an NTRK inhibitor that was approved in 2018 for solid tumors, another tissue agnostic approval, and then another one, entrectinib for NTRK fusion adult and pediatric solid tumors in 2019. And then another one, pembrolizumab for high tumor mutational burden solid tumors in 2020. But if you think that's the end, it's still going strong. And now we have belzutifan, a HIF inhibitor for adults with VHL with a variety of different cancers. And that approval just happened 10 days ago. And now we also have less than 10 days ago, 6 days ago, dostarlimab, anti-PD-1, again, for adult patients with mismatch repair gene defect solid tumors. So we now have 7 of these tissue agnostic approvals, and I think more are coming. So this brings me, because some of these are obviously for immunotherapy, I want to talk about genomics and immunotherapy, because they are really the pillars of precision medicine. And so you have genomics here and you have immunotherapy. And a lot of people think of them as separate, but I believe that they are a couple and they are married to each other. And what do I mean by that? There is a bridge between genomics and immunotherapy, and some of our most powerful predictors of immunotherapy response are genomics. So just as an example, the more mutated the tumor, the better the response to immunotherapy. And in our studies from UCSD, the response rate for low tumor mutational burden was 4%, 26% response rate to checkpoint inhibitors for intermediate, 45% response rate for high, and 67% response rate for high mutational burden. And so again, some of these patients, it wasn't so long ago when we saw these horribly mutated tumors that they were just chock full of mutations. We would have our molecular tumor board and we would hang our heads and say, we would feel sorry for the patient, because how are you ever going to treat a tumor that has so many mutations you can never target it? And then a few short years later, science changes that completely. And now when we see a tumor that is chock full of mutations, we are so happy, because these are the patients that have the most amazing responses to immunotherapy, some of them with long-term complete remissions, which I think may be cures. So as was mentioned, I founded a rare tumor clinic that turned it out to be a precision medicine clinic at UCSD. I've had a special interest in rare tumors because it's a huge unmet need. And also these tumors may be individually rare, but taken together, they make up about 25% of the cancer burden. So taken together, they're a huge unmet need. And what we did in all patients is comprehensive molecular profiling. That included DNA sequencing, protein analysis, immune signatures, liquid biopsies, and so forth. And we had a molecular tumor board that was attended by all sorts of specialists, our medical oncologists, surgeons, radiation oncologists, but also our bioinformaticians, our basic scientists, and so forth. And we did clinical grade molecular profiling on about 21,000 patients, a little bit more than that. And the idea here was, this was not an academic exercise. The idea here was to match patients with drugs. So we published in Nature Communications 715 patients that were brought to the molecular tumor board. And the bottom line is the results showed that the better matched the patients, the better they did. And each of these patients that were matched got tailored, customized treatment combinations, not just single doses. And I'm just going to give you some examples, and then I will go through some of the other studies that we did. This is a patient that I saw with a metastatic basal cell carcinoma. It's an ultra rare tumor. I saw the patient in the fall of 2015. He was a 55-year-old man. He'd failed multiple therapies. He failed 6 therapies, including vismodegib and sunitinib, which are the targeted therapies that target PATCH1 and hedgehog pathway that are FDA approved. So let's look at why he may have failed these therapies. And by the way, he was not in good shape. He was really headed to hospice. He had liver metastases, brain metastases, and bone metastases. And so what do you do next for this patient? Well, probably send to hospice might be the best answer, but we decided we should do, because that's what we do, comprehensive genomic profiling. And so this is his profile. This is a Foundation Medicine profile, 315 genes. And I think you can see immediately why he didn't respond to the targeted therapies that are targeted against PATCH1 or against that pathway, because he has so many other alterations. And so it makes it less likely that a targeted therapy against one of these alterations would work. We found that he had a high tumor mutational burden, very high, 79 mutations per megabase. And he also had PD-L1 amplification, which we later showed is associated with responses to immune checkpoint inhibition. Now, interestingly, the PD-L1 amplification was not associated with high PD-L1 by immunohistochemistry. I don't understand that very well. It may be a problem, technical problem with immunohistochemistry. But anyway, we went on and treated him with nivolumab, and this is his liver tumor pre-nivolumab. This is at 2 months. And the bottom line is that he has a complete remission ongoing at 5.5+ years. So this is something that we almost just never saw in solid tumors. It can be seen with hematologic malignancies where we can get complete remissions. It can be seen with testicular cancer, but other solid tumors, we've never seen responses like this, where a patient can go from having failed 6 therapies to getting an immunotherapy drug and having complete remission ongoing for close to 6 years. I'm very cautious as an oncologist of calling this a cure, but I think it actually is a cure. But I also want to tell you that it's not all good with immunotherapy. We all love immunotherapy, but we've also seen this phenomenon called hyperprogressors. And this is a phenomena where you give immune checkpoint inhibitors and the patient's disease accelerates. It's like pouring gasoline on a fire. And this is an example of a patient. This is 2 months before immunotherapy. This is a breast cancer patient. And then this is right before immunotherapy. So you can see in the 2 months, the tumor has grown a little bit, but not too much. And the patient gets nivolumab. And 6 weeks later, the patient has exploded. And so this is something that we and others have published. This patient had MDM2 amplification. And when we looked at our patients, there were 2 alterations that were associated with hyperprogression or accelerated progression. EGFR alterations, which have been previously associated with resistance, but also in our experience, about 20% of patients can be associated with accelerated progression. And MDM2 amplification, which we think is a very high risk for hyperprogression. Now, I want to emphasize that not all patients with EGFR alterations or MDM2 amplification will hyperprogress, but our data suggest that these 2 alterations are a significant risk factor. And I also want to say that there are patients that hyperprogress that have neither one of these alterations. And we also don't know why these alterations predict hyperprogression. So, but the important point here is that next generation sequencing, NGS, can help define not only who to treat, but who not to treat. And I think that may be just as important. So this is another example of a patient that came to our clinic, rare tumor clinic. The patient was a 49-year-old woman from Saudi Arabia. She had a high-grade neuroendocrine cervical cancer. She had been treated with surgery, chemotherapy, radiation therapy, and she really came in, it was January 2016, and she came in really lousy shape. She had this huge abdominal tumor, looked like she was 7 months pregnant, but it was all cancer. She had an impending bowel obstruction. She had an impending urethral obstruction. Our urology consultant didn't even want to put in a stent because he felt that she was, the only choice was really hospice for her. And so what do you do next for this patient? Well, sent to hospice is probably the most reasonable choice, but we decided to do comprehensive genomic profiling. And but we had a problem, and the problem was that we didn't have time to send her for a biopsy and get the next-gen sequencing back, because we felt that she probably was not going to live more than 7 to 10 days with all her problems. So we did something different, and that is a liquid biopsy. And this is how technology can drive even more findings. So we had experience now, by the time I left UCSD, we had experience with almost 6,000 liquid biopsies. You can do it on urine. Most of our experiences with blood, and that's what we got on this patient. And so it saves a lot of time. You just get a blood sample and you send it off. You don't have to get a biopsy. And this is what we found in her liquid biopsy. Now, having looked at a lot of these, we know that the average patient has about 3 or 4 alterations in their liquid biopsy, including deleterious and VUS. And here, this patient is chock full of alterations, over 20 alterations. Now, this is January 2016. So we had to make some assumptions, and we guessed that this was hypermutated ctDNA and that the patient had a high tumor mutational burden. So we went on and we treated the patient with, again, checkpoint inhibitor. This is her tumor before. This is 8 months later, and she went back to Saudi Arabia after she was in complete remission. And the last time I had contact with her was 3.5 years later, when she was still in complete remission. So again, what Dr. Sobeja has called these Lazarus responses, hospice to complete remission, pretty amazing. And in the meantime, we did her tissue NGS while she was getting treated. And our guesses were correct. The tissue NGS, this is again, Foundation Medicine. Usually you have about 4 alterations. She had, I think it's 23. She had a high tumor mutation burden of 53. It ended up that she was microsatellite unstable and that she had a mismatch repair gene alteration. And we now know that we can look at liquid biopsies, what we didn't know in 2016. And we can look at liquid biopsies and get tumor mutational burden, get MSI and so forth. So the important point is that new cutting edge technologies needed for complicated cancers and blood-based tumor mutational burden and microsatellite status is now available. And liquid biopsies are an amazing adjunct to tissue biopsies. But now I want to come to the final part, and that's what I started with. What if every patient with metastatic disease is different, like malignant snowflakes? And I'm going to give you this example. We've done thousands of samples with breast cancer, but they don't fit on the slide. So I'm just giving you an example of a series of patients done, again, Foundation Medicine. And I want to give you 2 patients to compare. This patient has ERB2 amplification, and this patient has ERB2 amplification. So obviously, both of these patients are going to get HER2 targeted agents. But according to our standard paradigm, they get the same regimen. And if you just look at what the molecular profile is telling us, they don't belong on the same regimen. Yes, they have something in common, and that's ERB2 amplification, but the rest of their molecular portfolio has multiple other alterations, and the alterations are different in patient 42 than patient 25. So we set out to try and do the next generation of studies was to customize therapy. So instead of using a consistent drug or drug regimen between patients, even in the presence of different molecular profiles, we used a consistent strategy, molecular matching, but allowed for different drugs. And so this is the N of 1 patient-centric trial. The drug regimen is no longer the center of the universe. The patient is the center of the universe. And you can see that patient 1 has markers A, B, and C, and patient 2 has markers A, E, and F. So yes, they both get drug A, but the patient 1 gets B and C with it, and patient 2 gets drugs E and F with it. And so that's the basic idea. And so we started this protocol that we called iPredict, and it was initiated in February 2015. we've now treated about over 400 patients on it. This is customized combinations. The first group of patients was refractory cancer, and we've published those. And the second group of patients is newly diagnosed patients with lethal malignancies. That data expands on our initial publication and is now in press. So the first publication was Nature Medicine, April 2019. And this is what we showed. If we look at clinical benefit rate, 50% of patients benefit. Most of that is complete or partial remission. The stable disease is at least 6 months. And that's if patients have a high match. If patients have a low match to therapy or no match, response rates are about 16%. Now, I want to I'm going to come back to this because this is very important. This means that patients that have a low match do, on the whole, almost as poorly as patients that are unmatched. And so I want to come back to that. And this is the overall survival curve. About 60% 2-year survival for patients with a high match, about 20% 2-year survival for patients with a low match. So the higher matching score translated into significant better response rate, overall survival, and I haven't shown it here, but it's in the paper, progression-free survival. So what do I mean by high match versus low match? Well, in its simplest form, if you have one alteration and you match that alteration, let's say you have the only alteration you have is RET, and you match it with a RET inhibitor, your matching score is 100%. On the other hand, if you have 10 alterations and you only match one, your matching score is now 1 out of 10 or 10%. So it's very low. So if you have 10 alterations, you really have to match a lot of those alterations to get a high matching score. And that's really where the customized combinations come in. So this is just an example of a patient. This is a 62-year-old man with poorly differentiated cancer of unknown primary. He has an KRAS mutation, which is not your regular KRAS, but it is pathogenic. And he received Trametinib, which is MEK inhibitor, and he has a RID1A, which we know sensitizes to PARP inhibitors, and he received Olaparib. And what you can see is he's had a very beautiful response to this customized combination with therapy ongoing at 15+ months. I want to finish off by just mentioning the study from WIN, our first personalized cancer medicine study through the consortium, whose mission is global delivery of precision medicine. And here the important thing is, I've talked about genomics, but genomics is just the tip of the iceberg. There's transcriptomics, proteomics, epigenetic changes, and so forth. And WIN undertook to do a study that incorporated not only genomics, but also transcriptomics. And this study, to my knowledge, first precision medicine study in solid tumors that included transcriptomics and genomics. And what we found was, not surprisingly, that transcriptomics was a really important addition in addition to genomics. We can't just rest our laurels on genomics. There's new trial types. I talked about real world data. We've initiated a master observational trial called ROOT, which takes real world data and turns it into structured data. So not just reading from the computerized records, which we know are difficult, but looking at structured data. And then just to finish off, what about the host? We've talked again a lot about the tumor, but I think we're reaching the point where we need to be doing germline on just about everybody, because the germline determines toxicity, the propensity to cancer. And here's this lady in your face at 100 years old, chain smoker. Is this good luck? Or is this genetics? I don't know for sure, but I'm betting on genetics. And then finally, the genomics immunotherapy era will revolutionize not just cancer medicine. And this is just something that really fascinates me. So we know that FGFR3 mutations are common in bladder cancer. We now have a drug to target it. But very interestingly, the same mutation in the germline, in the same gene and the same locus, causes achondroplastic dwarfism. And so that brings up the question, could we modify the phenotype of these patients with the same drug, with the FGFR inhibitor that we use for cancer? And I don't know the answer to that, but it has been tried in achondroplastic mice, and at least in the mice, it works. So I think this is going to spill over the genomic era into all of medicine. And so I want to thank everybody, and I'm happy to address questions. You're welcome to email me. I have a strange email address. This is my personal email address, and everybody, I've had a lot of questions. Where does it come from? I was reading in 2011, The Emperor of All Maladies, which is of course the Pulitzer Prize winning book on cancer. And so my email stands for The Emperor of All Maladies. Now, it's a phenomenal book, but the basic premise is wrong. The basic premise was that cancer is so complicated, it will never be overcome. And I have to say that I believe that reminds me of the 1990s, when we were told that the human genome is so complicated that it would never be sequenced. And I hope that just like that prediction was wrong, the prediction here in this book is also incorrect. And I think we are going to overcome cancer, and we're on that pathway today. So thank you, and I'm happy to entertain any questions.
Thank you. That's really spectacular. Thank you, Ray. For those of you who may not know, both Vivek and Ray, they are tumor agnostic, which I'm sure you must have learned the word agnostic by now, or tissue agnostic, which means they treat any type of cancer. They don't care whether a breast or prostate, as long as they have a mutation, they treat, or some kind of a targetable target. So. And another coincidence is, there are a number of coincidences being talked earlier. Vivek was hired to MD Anderson by Ray, or recruited to MD Anderson by Ray. So small world. So Ray, we have a number of questions in the chat as well as in the Q&A. There is one from Shibik Chakravarti Kannan. It's asking, MDM2 is very interesting. It's E3 ubiquitin ligase, targeting and causing P53 degradation. So gain of function of MDM2 may lead to the high TMB, I guess. So your comments.
So my comment is that the first thing is we do not understand why MDM2 is associated with hyperprogression. Our studies have clearly shown the association, and in fact, we initially had 2 patients in clinic that had very dramatic hyperprogression. They both had MDM2 amplification. And then I actually had called Dr. Sobeja for another reason. And I asked him if he'd ever seen hyperprogression. And his answer was, oh, yes. And I said, why haven't you published it? And he said, because nobody will believe it. This was in the area of the immunotherapy. And so I said, did you ever do genomic sequencing on any of these patients? And he said, well, I have one and I'll get back to him, but I didn't tell him about what we'd seen with the MDM2 in the 2 patients. And he got back to me and he said, the patient has he showed me the whole sequence, and the sequence included MDM2 amplification. And that actually gave me a shiver down my spine because it made me confident in our data. So I'm sure Dr. Sobeja remembers that conversation. The bottom line is, however, we do not understand how it works. So I have a collaborator at UCSD who was working on it in the lab. We don't understand the mechanism. That bothers me. I don't know that it's P53 because P53, to our knowledge, is not associated with hyperprogression. And so I cannot answer the question, which is trying to, I believe, get insight into the mechanism because we don't understand the mechanism. That it needs to be worked through.
Thank you. Thank you. So, there are a number of comments. Again, there is a question by Julia Kaufman. I hope I said the name right.
I think, what we can essentially do is we can jump on the panel discussion. This question exactly gets us to the panel discussion session. And I think the questions that we have, let's perhaps we can start with Dr. Kurzrak in the panel discussion. Let's see. The first question is, I think Julia Guffon is asking, in community settings, we often do not see diagnostic biopsies sent for the white panel NGS testing, perhaps only single gene testing in-house or PDL1 testing first. How can we convince these physicians of the power of testing upfront and not waiting for the patient to pass through all standard therapies first before testing? And, the question here is, Dr. Kurzrak, as we all, know, universal genomic testing upfront versus just hotspot testing in refractory patients. So I think it'd be great if you want to take on that question.
So I don't think that there's any question that every patient should have next-gen sequencing. And I think it's going to become standard of care. I mean, several panels are already FDA approved that are next-gen sequencing, but I think it's going to be as much standard of care as having the pathologist read the microscope. But I think one also has to maybe pay attention to why physicians are hesitant. And I will give you my experience because it was fed by coming to University of California, where you have academic physicians, but you also have a lot of physicians that are affiliated through practice. And what I learned is that some of the hesitancy to order the next-gen sequencing would not be intuitive to me. So first of all, they were afraid that the patient would have a big charge, like $5,000 or something. And then and the second thing was a lot of the panels are very complicated and they're hard to interpret. And I think they didn't say this, but I think that they didn't want to look foolish in front of their patients. And so having a molecular tumor board was very helpful to the physicians because they had some place where they could go and ask questions. But I think those two issues, it's not just about education, it's about the business aspects. The first worry the physicians had is there may be a big charge and then they have to deal with an angry patient. And the second worry was knowing enough about this to be able to, or having somebody to ask, and that was the molecular tumor board.
All right, outstanding. I don't know if Dr. Janeway is here. Maybe we can ask the next question to Dr. Janeway if she's here.
Dr. Janeway, are you here? She's on the call.
Yeah, she's probably on mute. So maybe the next question can go to Dr. Susan Murray. Manny, do you want to go ahead and ask that question?
The four, yes, I know that.
Yeah.
We have a set of questions prepared. That's why we are just saying numbers. So, Dr. Murray, so precision medicine, it seems like a, very, it's a niche, a boutique initiative in the major centers only today. So there was a recent report from the VA that not many patients, even the lung cancer patients, are getting the standard of care. It's just follow-up of the previous question, standard of care NGS. So how can we improve upon precision oncology education, particularly making everybody understand that NGS should be part of standard diagnostics? so that people don't have to wait until the doctor says, oh, you are not responding to X, Y, Z, therefore now you should get an NGS. So we'll see what will you be, what kind of treatment option you may have. It's similar to what the question somebody asked earlier. Your comments, please. How do we educate?
So from an education perspective, I have two responses. If there is an opportunity to do a much more rigorous study to assess the causality, why are they not? Why are they might have the knowledge, they might know they should be doing it, but assessing the causality of why they are not doing it. So it might not be that they don't know, there might be other causalities, right? There might be other issues, whether it's their level of confidence or they're not necessarily comfortable, or they don't have the system or the team to support them in that decision. So that's what we call more rigorous ethics approved needs assessments. Those are studies, and there are many publications out there in relation to needs assessments. And that's the more scientific approach, where you would conduct in the institution with an appropriate targeted audience, appropriate subjects, those that you wish or hope they should be, as we were talking about gap analyses earlier, that you'd be able to assess why are they not doing it? And then once you have the why, if it's knowledge-based, then obviously your educational content needs to be looking at addressing the knowledge, but in their context. How do they apply it in their clinical setting, in their particular, with their particular patient profiling and so forth? So that's the rigorous approach. If you don't have that opportunity, then obviously it's not necessarily optimal to just tell them to do this. Adults don't do very well with being told to do something. So again, using what we call case-based learning, so having multiple different types of cases, preferably cases that you can identify from their practice, from their perspective, because they might not be experts, they're like yourselves. So obviously getting what are the challenging patient profiles for them? Why? Again, we're back to the why. So those are really, from an educational perspective, is you want to get to the why. Why are they not doing it if they do have the knowledge? And then you target your content and the instructional design to the why. So this way you get better probability of them actually changing their behavior. Is that helpful?
Yes, thank you. Thank you. That's, yeah. So, basically making everyone aware the benefit, the power of this. Again, as Ray was mentioning earlier, the cost is coming into the picture. That's where cost versus benefit. And here is Dr. Bricker, while she was debating about a cost, but cost versus benefit, she went for a benefit rather than the cost at that point. That made a huge impact in her life. And so it's very important to keep that in mind. But, in the US, who is going to pay for that cost, right? Is it a patient or the insurance company? That's where things get mixed up. But anyway, I think Vivek has a question after me.
So great. So again, that reminds me of the next question is, again, maybe Dr. Kazra can tackle this. So even with the best of targeted therapy, ultimately we see resistance. Resistance is a rule rather than an exception. And of course, we completely agree that combination therapies are needed, but, these have been, very challenging in real world scenarios. So what adjustment in the clinical trial design and, regulatory and care settings are needed to move from a drug-centric to the patient-centric approach that you mentioned? The challenge is, how do we access these drugs? Because, these drugs are approved for different indications. So, how can real world clinicians overcome these hurdles?
Right. So I think you're asking maybe the key question. So when we started the first, to my knowledge, customized combination therapy in early 2015 at UCSD, I want to say that it took us 2 years to get it through the IRB. And I think we're fortunate that we got it through the IRB at all, because we were going to give, for the first time, individualized combinations. But one of the things that helped, and actually gave me insight as well, was thinking about how, as a physician, we practice and understanding that giving customized combinations, not giving customized combination in oncology is the exception to the rule rather than the rule. So the average patient comes in to the oncology clinic on 8 different drugs for their comorbidities. And you and I and every oncology physician, as well as every physician, give customized combinations of drugs to patients every day. If your patient has heart disease and an infection and rheumatoid arthritis, they give a different set of drugs than the patient that with depression a bowel disorder or something like that. So we customize every day. Only in oncology do we not customize. And I believe that was the legacy of the cytotoxic era. So the first thing we had to do is a trial to show that it was safe to do this. Now, remember, I'm a phase 1 physician. But here we were doing a trial without phase 1. And everybody would get a customized combination, no prior phase 1. And what we did is we followed some rules, but the bottom line is that we lowered the doses to start with in every patient. And every patient, then we followed them carefully and basically customized the dose, but we started with a lower dose per patient. And we were able to show, and this is in our publications, that our serious adverse event rate was less than with standard of care therapy. So the first thing you have to address, number 1, is the safety. And I think we've addressed that. Now, the second thing that we had to address is actually getting the drugs. And for the trial, we had a certain methodology to get the drugs. I joked that we hired a magician, but it was really a very dedicated human being that was a medication acquisition specialist and also a clinical trial navigator that would help navigate patients to the right clinical trial or get the medications for us without charging the patients. And there are lots of mechanisms to get medications without charging patients. But what works for a clinical trial at one university is not scalable for the country. And so we felt that our job was to prove the point. And then the next level will be, honestly, for the FDA to accept some of the algorithms that maybe we used. And if that becomes accepted, then it becomes acceptable to give these customized combinations to patients based on the algorithm, based on the strategy, rather than based on the impossible approach that we've had historically, which is to try every combination in every different disease over and over. That just can't work. You have 4.5 million combinations for 300 drugs. That's not happening. There's not going to be that many clinical trials. So I think once we've done it in the clinical trials, the algorithms will at some point have to be looked at by the FDA.
Absolutely. Again, I wanted to ask also this question, feedback to our FDA speaker. I don't know if Dr. Drezner is here. Dr. Drezner, are you here?
No.
No, I think she had to drop off for another FDA meeting. As you know, FDA is, it's been a historic day in FDA with the first approval of COVID vaccine. Everyone's busy. We thank.
I have a question for you, Vivek.
Yeah.
I have a question for you. Again, you being a pediatric oncologist as well as treating adult cancer. The question I have for you is, should all children with cancer undergo germline testing? So they probably need to get whole genome testing as our hotspot panels may pick. So in other words, should we do,
Dr. Janeway is here. She's the perfect person to answer this question.
We were going to ask you that.
Perfect.
So, should all children with cancer undergo germline testing?
Yeah, this is a really, really good question. So I think it depends on the disease subtype they have. I am primarily in the extracranial solid tumor and sarcoma clinic. And for our sarcoma patients, we are actually referring almost all of our patients to cancer predisposition clinic. The literature basically suggests that the frequency of germline cancer risk in pediatric cancer patients is about 10%, so 1 in 10 patient, very similar actually to what's seen in adult oncology. But we approach it in a similar way as for adult oncology, which is to send those patients with the disease types that are most associated with risk of germline cancer predisposition. That said, I think from a research perspective, we at our institution will often have a research protocol in which each patient can enroll, because I think there's still so much to be learned in the cancer predisposition space in pediatric oncology. So we try to encourage, outside of those disease types where it's indicated, enrollment on a clinical research study.
Thank you. I wouldn't have expected that simply because one would assume adult, the germline may play a role, but maybe to a lesser extent than a children, because they had a limited amount of time with all the bad chemicals and whatnot. But it's surprising to hear that both in adult as well as in children, the germline mutation causing cancer is like a 10%, if I understood correct what you said. That's remarkable.
So let's have probably Dr. Kazrak to comment on that. I think we may have some, other views, but Dr. Kazrak, you can comment on that.
Yeah, so Dr. Sobeja and I have talked about this. I think we have similar views. I'm actually of the opinion that every cancer patient should have germline testing. We know about 10% of adults have germline. I have repeatedly seen patients whose cancers don't apparently read the genetics book. One of them, actually, I remember seeing when I was still at MD Anderson a decade ago, 72-year-old man, had somatic P53 mutation, no prior cancers, no family history of cancer. We had him on a research study. He had germline P53. Obviously relevant to the family. And we've seen this repeatedly. I think it's only a matter of time until germline testing is really done on every cancer patient. 10% is a lot of cancer patients to have germline. And I will say that even if it's rare, and maybe that I'm a fan of rare things, but just even if it's 1%, we in medicine, we don't want to miss 1% of problems. We don't even want to miss 0.1% of problems. So I do think that this is going to become, germline is going to become routine at some point. I realize not yet, but that's where I see the future going.
I was just going to say, we don't disagree at all. All of my patients see, I can answer what I do. I send all of my patients to the cancer risk clinic, and we have an active research study. I think if you asked me in pediatric oncology in general, what's happening, that was my prior answer.
No. I totally agree. And I was trying to give that it's not just pediatrics. I mean, you think of pediatrics right away because children aren't supposed to get cancer. And you sort of, you look at a 60-year-old or 70-year-old and you say, well, they're old, they get cancer. But I don't think it's that simple. And the germline may be very relevant to their family. So just because somebody's older when they get their cancer doesn't mean it's not germline.
Thank you. I just have a question for Vivek and Ray. It's a comment from the chat. One by Zakia, I cannot say the last name, and the other one is from Tofik Aditro. And they're asking, sometime mutations are not the driver of the cancer, right? How do you determine that to be sure that we offer the right therapy under that circumstance? What about other way of profiling? And the similar question is if there is a patient who was on a personalized therapy or precision medicine, and if they don't respond to it, which is kind of very similar to if it's an oncogene is not a driver, then how do you treat?
Yeah, go ahead, Dr. Kajak. You go ahead and take that.
Yeah, so, I think the science is still incomplete. What we've learned is that some types of alterations are more likely to be drivers than others. And like one example I'll give is fusions. So when we see fusions, we believe those are very strong drivers, and we've published on that. And we've even shown that if you have a fusion and you have other alterations, if you target the other alterations, but you don't target the fusions, it's almost as if the patient was unmatched. So what we do is, sometimes it's hard to target all the alterations. We target as many as possible in our customized combinations. And then if it doesn't work, we try a different customized combination because our science is not, we're on the path, but we don't know everything about every alteration. So I think with time, as we learn more, we will learn more about like a fusion is more important, or that certain alterations. And I alluded to something else before, and I want to reemphasize it. So far, all we're doing is genomics. We're not doing transcriptomics. We're not doing proteomics. We just published a paper showing that about 15% of driver genomic alterations are not expressed. They're silenced at the transcriptomic level. So if you have a driver and it's not expressed in the RNA for whatever mechanism, that's going to be a resistance mechanism. So I think we try another customized combination, but I think what is the most important thing to realize is that the science is moving quickly, but there's still a lot to learn, and we're still at the tip of the iceberg.
Thank you.
Yeah, and as I showcase here, Dr. Kajak and I wrote this some time ago, and this is what we do. I think with genomics, we are just touching the leg. All right, we need a comprehensive molecular portrait of the tumor. We don't know much about metabolomics, transcriptomics, immunogenomics, and microbiomics and proteomics and beyond. So I think we need a panoramic of a molecular portrait of every patient with cancer walking in the door. And just as an aside, Dr. Kajak wanted to make sure that it's balanced among men and women. There were 6 blind men and women scientists and the elephants here. And it was like, this was a long time ago. So, she wanted to always make sure that, there is balance and equity in science. Thank you. And, it so happened to be an all-woman speaker panel today. And, we didn't plan that, but I think, this turned out to be a rock star panel, right? Including from FDA and among others. Thank you so much.
Thank you. So I have a Vivek, do you want to comment that? So you already commented, both of you commented on the same topic. Thank you.
Yes.
And you have
so I think, in the interest of time, maybe let's have, the final question for Dr. Bricker. I think, thank you so much, Dr. Bricker. Ali, I think that was a powerful presentation. It gave so many of the participant goosebumps when you showed those slides. It was very powerful. Thank you so much. We wish you good luck. And one thing is, how can what do you think about the role of social media in patient education about NGS testing in patients with cancer?
I think that the message should be blasted far and wide that every patient should get NGS testing. Initially, all of my markers came back negative. So if it hadn't been for you picking up the phone and calling me, I would have just gone along with that. And this is the United States. I think right now, it's not 2015 anymore. I think at most academic centers, most cancers are getting sent for extensive biomarker testing, but what's happening out in the community is a totally different story. And just from our Facebook groups and from Twitter, we see all around in different parts of the world, there's quite a lot of resistance to testing people for these biomarkers. And in many cases, if you find it, you can treat it. But if you don't look, you'll never find it. So.
Well said. Yeah.
Yeah, thank you so much. I think, people always say buy the genetic lottery. Of course, unless you buy the lottery, you won't win the Powerball.
Vivek, we do have some more time.
We have some more time. Great. Wonderful.
12:20 to 1 o'clock was the panel discussion.
All right, perfect.
And we are 20 minutes up into it. So I have a question to Dr. Janeway. See, the thing is, we are, as some of you touched upon earlier, we are just looking at a genomics. And as we have various omics. On top of that, there is a single cell omics and spatial transcriptomics, and where you have a particular cell sitting next to another cell, that makes a huge impact, right? And while those technologies are like getting developed, particularly to understand what immune cell is telling the tumor cell rather than whether immune cell is next to the tumor cell, and the vice versa, that communication. We would be able to understand a lot through spatial transcriptomics, for example, or multi-omics. So what do you think, in your opinion, should energy and effort should be spent extensively on those areas to understand that, or should we just focus on total transcriptomics to start? Because from genomics, the next level should be total transcriptomics, and then see how that would help treat patients.
Yeah, I think, so I love that elephant figure. I remember seeing that at one of the conferences, and congratulations to you for including that gender balance. I really appreciate that. The way we're thinking here is for some entities, for right now, the genomics is maybe sufficient, but for some other entities where the genomics are less revealing, and when I say genomics, I mean DNA and RNA, but RNA mostly looked at for fusion, with the occasional use of RNA for sort of outlier expression. It's very difficult to use clinical samples, so FFPE embedded samples, to look at sort of transcriptomics in terms of gene expression. It's just, there's a lot of artifact introduced by tissue processing. So I think for some of the diseases we treat, it's not adequate forever, but they are common enough that we can leave the sort of discovery of the other omics to sort of the standard research pace that happens in people's basic science laboratories, because the DNA and RNA omics provide us enough information to take care of that patient adequately. I do think that there are subsets of cancer types where those of us who work between the clinic and the laboratories and have really pushed precision oncology need to start to think about multi-omic profiling in a way that we can actually use that information in treating the patients. In my ideal world, we would do it with every cancer type, but it's too much information to be able to, process and analyze that information in a way that's helpful for the patients if you just do it for everybody. So I think there are some cancer types where we should focus on getting multi-omic data in sort of as much real time as we can. For me, in the world I live in, the cancer type where that I think is most important is undifferentiated sarcoma, where people have started to look at where we don't even know what to call the cancer. I think in those cases, it's going to be important to start thinking about sort of a real multi-omics approach. I'm not saying it shouldn't be done in other cancer types. I think it will just be done in those cancer types in more of our sort of standard paradigm of, somebody's really interested in the microbiome of the tumor, they'll do that. Someone's really interested in immunotherapy, they'll look at the sort of relationship. The last thing I'll say in response to that question is the thing about the elephant that's so good and is illustrated so well is one of the biggest challenges is kind of, even if you do each of those things, analyzing all of that data together and putting it together is really very challenging. So, and what's not, I don't know, maybe you can put it back up. I can't remember if it's there, but we also have started to think about phenomics, and I really have to give Dr. Deb Schrag, who's here and well now at Memorial Sloan Kettering, but was here, for sort of coining that term and helping us think about that term, which is sort of how does the patient, what are the clinical features of the patient, and how do you capture those clinical features in a way where you can do it similarly across institutions and share that information to sort of learn more about rare cancers. I don't know if that answered your question, but that's sort of how we're thinking about it.
Thank you. Basically, there is a laboratory science, there is a clinical science, and this should be part of the laboratory science. But as and when things evolve, should get moved into the clinical space and to make a next set of decisions. No, that's wonderful. Thank you. And if you all could just take a look at this chat as well as Q&A, there are a number of questions. If you could respond to some of them, will be spectacular.
Could I make a comment
please,
about the previous question?
Please.
I just want to a little bit expand on what was said, and that's about immunomics. And to me, this is a really, in addition to, like transcriptomics or proteomics, is looking at the immune environment. And at UCSD, we used an assay called OmniSeq, which has about 50 immune markers and clinical grade. And what I think is remarkable about this is that immunotherapy, maybe because of the big successes with the anti-PD-1, did not learn the lessons that we learned in genomics. So now we have drugs like, let's say, IDO inhibitors, and they're not doing well in the clinic, or they fail in the clinic. And then we can ask, well, why are they failing? Well, when we looked at IDO overexpression, it's about 5% of tumors. So if you have 5% of tumors and you don't select the tumors, it's the same as giving an ALK inhibitor, almost the same percentage as giving an ALK inhibitor to lung cancer, and ALK rearrangements occur in 4%. If you give it to every lung cancer patient, ALK inhibitors fail. So I just wanted to put in a plug for immunomics, or at least looking at markers. OmniSeq uses RNA markers, RNA expression markers. But we have hundreds of thousands. I think it's estimated that the clinical trials in immunotherapy will include 380,000 patients. And almost none of them have any biomarkers. And so I think that's a mistake. I think we are going to discard important drugs. And even if we have drugs that go through for FDA approval, surely we could get better responses if we could narrow the population into the ones that really need the drug. So I just wanted to put in a plug for immunomics.
Absolutely. I think understanding all this together is what is going to be an ultimate. But again, bioinformatically, putting all this messages, information together and make it understandable to those who are trying to practice, or for anyone, it's going to be really challenging. But thank you. That's a very important point.
So, again, as we know, there are some groups of people that say that precision medicine would never work. Again, today we had the historic approval of COVID vaccine, full approval of COVID vaccine. As I mentioned earlier, it's going to be called Comirnaty. That's a name. I don't know how they come up with names, but it's Comirnaty. It's going to be a household name in the near future. So COVID vaccine is ultimate precision medicine. It works. COVID vaccine is a designer mRNA for the spike protein of coronavirus. I'm sure this technology is going to take off for patients with cancer. And COVID monoclonal antibody is also precision medicine. It's again against a biomarker. Again, what we are looking in the future is COVID taught us a lot of common sense issues. Patients on oral TKIs, we could ship the drugs. And when we designed clinical trials, it was more of patients being the trial being clinical trial centric. Hopefully the COVID-19 pandemic and the cancer clinical trial pandemonium will find a silver lining in the COVID pandemic from moving from clinical trial centric to patient centric model. And one thing is everyone was asking about the videos of this, so we will record this and share it.
Record it. I'll just show this, Vivek. This you can find very soon. We will share the link. Are you able to see my screen? No,
not yet.
Okay, sorry, I'm trying to share my screen. Okay, share. So it'll be shared and in the Twitter account of both Vivek's and mine, because I don't know that this is where it'll be shared.
No, so it says no. It's zooming in on something else.
Got it. Okay. You can how do I? This is okay. Stop share. You can share it if you have it. So it'll be shared on our Twitter account. Now I'm really motivated by Dr. Bricker that I am going to use more Twitter than before. It's definitely, that's a place now. Social media is changing everything. So we'll post stuff on a Twitter as well as LinkedIn. Then you all can use it and pass it on to others. Yep, there you go. Thank you. Yep. Yours works. Wonderful. So again, I don't know whether Vivek, you have anything else to add. We are right on time, even with a small glitch in our planning. But thanks to the speakers for accommodating. And some time ago, I hosted another meeting where by accident it happened to be men, and then people commented that I held a meeting with only men in the panel. It was not planned. The women speakers who I invited didn't accept. They were busy. So now I'm happy. Now this is all women panel. So now I've just compensated for that. And then we really appreciate every one of you for making your valuable time to come and speak and educate others. And Vivek, I think is going to provide the concluding conclusion. And
thank you so much, each and every one of you, for taking their valuable time off and being for the event. Thank you so much, and thank you for the audience, and thank you, Dr. Mani, and thankful to Ebony, and she's completely remote, and
very important.
She's fantastic, because again, I'm very bad in answering emails. Dr. Mani knows that, and I respond after especially from an inpatient service. So Ebony chased us to make sure that this happens. She locked in a time, date. We thought COVID would be over by now, but unfortunately, Houston, Texas is now become the epicenter of COVID pandemic, and we're all being pulled to cover services. So glad that we could do this virtually, and I guess we got audience from everywhere. Almost every continent. I think every continent we got people from Asia, from Australia, from Africa, from Europe, from Brazil, and the US. So I think we covered all the continents except Antarctica. So that's the beauty of virtual Congress meetings, is we can do this. And I'm sure next year this is hopefully can be in person. Hopefully the COVID vanishes from the face of the earth, and we can all smile at each other in person. Right? Thank you so much. And Argentina, someone from Argentina. Thank you. Thank you for joining.
Thank you all. Thank you so much.
Thank you.
Thanks, everyone.
Yeah, thank you.
Thank you.